You can see my screen, right? Online students, people who just joined, this is sort of the warm-up paper that we are discussing today. These are questions in the tute, right? After the microscope. Easy questions. It's fine. Just listen to the discussion, okay? Okay, 2 more minutes, online students. 2 more minutes. Did you think like today is a holiday? No, right? එහෙම කතාවක් තිබ්බේ නෑනේ නේ? It's a holiday again, right? Because we have 5 days, 5 Sundays this month. No, right? Yeah, so we are trying the questions after the microscopes. Just for MCQs, quickly try again. Can you hear me? Yeah. Online people? Okay. I will start the discussion. People who just joined, we are going to discuss the MCQs are from microscopes that is in the tute. Okay. So you can hear me and see me clearly, right? Yeah. Okay. So first question, question number 3. That is, so usually if they are getting question from microscope is going to be this sort of number, right? So first question, if there is one, it's going to be from introduction to biology, right? Sometimes there can be one, sometimes there can maybe not, right? But it's like year after another, there's question from introduction, then there's not. So usually, so MCQ number 1 is from introduction. MCQ number 2 would be usually from your biological molecules, right? Usually there is definitely one, okay? Then third one is again a bit variable. Microscopes වලින් අහන්න පුළුවන්, maybe not, right? But if there wasn't a MCQ for microscopes, there's going to be a small part in the structured essay. Okay, එහෙම තමයි pattern එක. So usually question number 3, O4 is from microscopes, right? එතන ඉඳලා පටන් ගන්නවා cellular basis. 4 onwards is going to be cellular structures. Okay. We're discussing these four MCQs. Okay. Right. Okay. Question number 1. Online students, any questions? Steps involved in observing an onion peel mounted on a glass slide and placed on the stage of a compound light microscope are as follows. A. Adjusting the mirror. B. Use of fine focusing. C. Use of coarse focusing. Okay. So I hope you saw those in like how it's done in real life, right? So without like before doing anything in a light microscope, what do you need to have? You need to have light. Light illuminating the specimen should be there. No? So what is definitely the first step? You have to adjust the mirror, right? Without light reaching the specimen and going through it, you can't see anything. So first of all, you have to adjust the mirror. Whatever, right? So they are implying this is a mirror microscope, like the one I brought, very old one, right? So it could be either switching on the light, something like that, adjusting the mirror. Use of fine focusing and coarse focusing. Which one comes first? Online people? Which focusing do you have to do? Yeah, you have to do coarse focusing first, right? Roughly, අඩුම ගානේ පේන level එකට ගෙනල්ලා තමයි you have to like nicely fine tune it, right? That's how you do it. No? So coarse, using the coarse focus, you have to at least make it a little bit clear. Then you have to make it very clear so you can see the details. So coarse focusing first. Usually the coarse focusing knobs locks. You can lock it, right? You can lock the coarse focusing knob. Then you will be able to rotate the fine focus, right? So both at the same time, සාමාන්‍යයෙන් in a good microscope ඔය දෙකම එකපාර use කරන්න බෑ. After using the fine focus, you can lock it. Then you are able to rotate the fine focus. එහෙමයි සාමාන්‍යයෙන් in a good microscope it happens, right? So A, C, B is the answer. No? Answer number 2. Okay. All right. Next MCQ. In a compound light microscope, the lenses reflect light to magnify the image of the specimen observed. What's the issue with that? Is there an issue? They are asking the correct statement, right? So හතරක් වරදින්න ඕනේ. So what's wrong with the if there's something wrong, what's wrong with the first one? Okay. Anyone else? What's wrong with the first statement online people? Yeah. Carefully read it. මොකක්ද අවුලක් වෙනවද මේ statement එක? මම කියනවා අවුලක් තියෙනවා. Yes, there's an issue. What's the issue? Lenses reflect නෙවෙයි නේද? They refract, right? A lens can't reflect. A mirror can reflect, right? Lenses bend light, right? Based on what, like our density difference, right? There's like refraction, so light rays slow down and they bend. No? I think you will do so many questions from refraction in physics. Okay. So that's wrong. So you can write a small review, no? Or you can cut the word reflect, right? You can cut the word reflect and correct it. Lenses refract light to magnify the image of the specimen. Online people, do you get it? What's wrong? So you have to be very careful when you read statements. Okay. Second statement: resolution power is inversely proportional to the wavelength of light. Resolution. It's a measurement of what? The clarity of an image. You know. Resolution power. Ability to resolve. Right? See two things as two things. Right? For someone who is at the very back, these two dots might look like one dot, right? So that is because as the distance increases, they are like our eyes' ability to separate two things. I mean, that is related to the clarity, no? So how clearly you are seeing two things, right? So this resolution power depends on what? Right? It's actually one of the biggest limitations of light microscope, no? So the resolution power is inversely proportional to wavelength. Yes, when wavelength increases, the resolution power decreases. Yes, this is the logic, no? So what do you mean by wavelength? It's like the distance between these sort of, what are these, strophes and the wavelength of parts. I can't remember. These are like peaks and these are like troughs or something. So the distance between these two kind of things is the wavelength, right? I can't remember. This is lambda, right? Something like that. So that's why we started using electrons, right? That's why we started using electrons, because electrons have a lower wavelength. Electrons have a lower wavelength, so the resolution power increases. Okay. So resolution power is inversely proportional. Yes. You can just we wrote this down, right? If you haven't, you can write this down. Resolution power inversely proportional. Right. Third statement: the image produced by the eyepiece lens is magnified by the objective lens. Who is doing the magnification first? You have to think from bottom upwards. No? That's where the light is coming. Light originates from the bottom part of the microscope, either the mirror or the lamp. To reach your eye. So through the specimen, the light reaches first what lens? The objective lens. So objective lens magnifies first. Okay, objective lens does the magnification first. Then those waves will reach the eyepiece, and the eyepiece will magnify it for the second time. Okay. So what's wrong? The image produced by the eyepiece is magnified by the objective. No, you can correct it with pencil or something. The image produced by the objective is magnified by the eyepiece. Image produced by the objective is magnified by the eyepiece. And last week, I sort of drew a diagram that is not very accurate by physics, right? So that's a bit embarrassing. So I thought of giving you the proper one because you will learn about it, right? I gave you a bit of a wrong line diagram last week. So when you are using a microscope, right? So if this is the object, the specimen, right? Your rays sort of come like this to the objective lens, right? And here you have what? Let's say the eyepiece. Okay, these waves are sort of reflected like this. Okay, they go like this, and the other waves sort of go through the lens like this, and they are supposed to sort of meet over here, right? They are sort of supposed to cross over here. Okay, and so I'm not a physics person, but this will be the view, right? This is how you are going to see the object, not like the one I drew, right? So this is the objective lens. This is the eyepiece. Okay. So the actually the what we call as the object. Sorry, the image produced by your light microscope is actually what we call as a virtual image, right? The image you can't project it to a screen, right? These are things you will learn under light in physics. Okay. So the image is actually produced over here, right? A massively magnified image will be produced sort of over here, right? You will need to actually memorize these line diagrams, right? No need to like just take it down with the pencil. This is the proper one. Okay, what's happening in a microscope? Okay, this is the proper line diagram. Just a rough idea. Okay. So this image is actually not a real image. It's what we call as a virtual image, right? Virtual image. You can't take it to a screen. We only can see it with our eyes. Okay. Using instruments, we can produce two types of images. We can produce real images and virtual images, right? So the image produced by my projector lens, you can take it to a screen. So that's a real image. Okay. No need to sort of pay this that much attention. Just I'm sort of when once you did lenses and go back to this sort of tute and you'll see make one right. So okay. Okay. Statement number 4. The maximum magnification. The maximum magnification is usually 600 times of the actual size of the specimen. We are talking about compound light microscope. So in the compound light microscope, what is the usually maximum magnification? We have the eyepiece that is about a fixed magnification about 10 times. We have usually 3 choices of objective lenses for 10, 40, 100. One is in a proper microscope, right? So if you choose the highest power objective lens, you get a 1000 magnification usually, no? From a light microscope. Okay, so that's wrong, no? You can correct it. Usually 600 times no, about 1000 times. Resolution power is 0.2 micrometers. Sorry, millimeters. Very sorry. So is it 0.2 millimeters in a light microscope? No. Even from our naked eye, we have a resolution power of 0.1 millimeters. Right? What's the resolution power of the light compound light microscope? It's given in the resource, right? 0.2 micrometers, I think. Yes, micrometers. Is it 0.2? I can't remember exactly as well. 0.2 micrometers. Check the resource book. Yes, resolution power is 0.2 micrometers. Okay, so you can correct it in the tute. It should be your review. So the resolution power of the compound light microscope is 0.2 micrometers. Okay, not millimeters. Right. So second MCQ done. Any questions? So the what's the correct one? The statement number 2 is the answer. No. Right. Next question. Which of the following statements is correct regarding transmission electron microscope? So what should come to your mind when you think of the transmission electron microscope? The electrons go through it, right? Go through it. We use it to observe inner structures, right? Because we are going to slice it very thin. The specimen, we are going to slice it very thin, and we are going to send the electrons through the specimen, right? Scanning it bounces over the surface, right? Bounces over the surface. So transmission specimens are magnified 5 to 10 to the power 6 times. Laksh Pahak, right? 500,000 times scale again. So what's the magnification given in your resource? Is it given? Electron microscope? Yes. In practice, page number 23, it says, in practice, electron microscope magnifies about 5 into 10 to the power 5 times, right? Not 5 into 10 to the power 6 times, okay? So, electron microscope magnification, EM, is what? 5 into 10 to the power 5. So, 500,000, right? 500,000 times. So they are given as 10 to the power 6, no? So they are saying it's 5 million times. Okay. So no, not that much. Only 500,000 times. So first statement wrong. Less electrons may get displayed in cellular structures with dense staining. Right? Stain is designed to do what? To attract electrons, right? So if a structure is densely stained, it is going to get more electrons attract. They get more electrons displayed. right? So if a structure is densely stained, heavy metals will end up stained, so heavy metals attract electrons, right? So the structures that are densely stained actually attract more electrons. Okay, so it's wrong. Or you can correct it by cutting the less electrons and saying more electrons, right? More electrons get displayed, right? Uh, Living specimens cannot be observed. Living specimens cannot be observed. True, no? Because two things. The electrons are going to kill the specimen anyway, right? And the other thing, when you are preparing the specimen to be observed under the electron microscope, it's going to kill the specimen as well, because we are going to use heavy metals, no? Heavy metals දාලා මේ stain එක කරනකොට anyway we are going to kill the specimen, right? For example, if you are observing that onion peel, it's going to get killed, right? Electrons යනකොටත් it's going to get killed, and when you are preparing also, it's already dead. Okay. Living specimens cannot be observed. Yes, that's the statement, correct statement. Fourth one, three-dimensional appearance of specimens can be observed. 3D appearance. Yeah. කාගෙන්ද අපිට 3D බලන්න පුළුවන්? Scanning එකෙන් නේද? So can be observed, you can in front of it, right? Using scanning electron microscope, right? In front of the fourth statement, right? Yes, 3D appearance of the specimens can be observed using the scanning electron. Okay, not the transmission. Specimens scatter many electrons while others are absorbed. Yes. This scattering of electrons is done by what microscope? Electrons are scattered. When the electrons hit the specimen, some are absorbed, some are scattered, reflected back. That is in the scanning electron microscope, right? This scattering happens in the scanning electron microscope. So what happens in the transmission is when electrons pass through the specimen, more electrons get displayed in regions where the structures are densely stained. That's what happens in the transmission. But in the scanning, they get some are absorbed and some are scattered, reflect, right? Okay. You can, in front of it also, you can write this happens in the scanning electron microscope. The last statement, the scatter many electrons, that happens in the scanning electron microscope. Okay. Okay. Last question. Which of the following statements regarding microscopes is correct? Right? Correct. In a light microscope, visible light is passed through the objective lens and then through the specimen. Right? Specimen එක through ඒවල තමයි objective එකට යන්නේ, right? Through the specimen objective eyepiece, then your eye. That is the pathway of light. So you can correct it. Visible light is passed through the specimen first and then the objective lens. Final MCQ. Okay. Second one, projection of light beam through a vacuum is the principle of an electron microscope. Through a vacuum? Yes, I think it happens in a vacuum medium, right? Let's keep that. Let's see if there is a better statement. Usually we need to have a vacuum for the electrons to sort of travel without any obstruction, because they can get absorbed into the air as well. atoms atmospheric air, right? So those can also get the electrons can get absorbed to those as well. Let's keep it. Second one, projection of third one, scanning electron microscopy used to study the internal structure of cells. No, to study internal structure, we need someone going through, no, transmission electron microscopy. So you can correct it or cut it and say transmission electron microscope is used to study internal structure of cells. Transmission electron microscope is used for detailed study of living specimens. Nothing living can be observed. Okay. You can either you can say you can cut it and say light microscope is used for detailed studies of living specimens. Okay. Or you can cut the living and say transmission electron microscope can be used for non-living specimens. Either two ways you can correct it. Right? Just get into your heads the logic. That's enough. Right. So I'm giving you the sort of freedom to sort of work with the tute as you like, because at the end of the day you are going to be studying it. Right? But correct things, okay? If you have doubts, you can ask me, right? If you ask your friends, if the friends don't know, then ask me, okay? Simple, right? Last one, magnification and resolution power are important properties of all microscopes. Yes, right? Magnification and resolution power are important for all. Second statement, projection of a light beam. Second statement, nobody told me. Projection of a light beam, electron microscope, a light beam. So definitely second statement wrong. What is it? Projection of an electron beam. You can just cut it and correct projection of an electron beam. No? Through a vacuum is the principle of the electron microscope. Like clearly the last statement is the correct one. Magnification and resolution power, important properties. So what is the formula for magnification power? Yes. The formula again. So resolution magnification is image size, okay, divided by object size. So in this here, this is the specimen or the object. This long thing is the image, right? So can you see, using the two lenses, we have increased the size of this to about this. That's what you need to do in a microscope, right? ඔගොල්ලෝ මේ වගේ line diagrams and the ray diagrams, right? For microscopes and and compound microscope, and also for the simple handheld lens එකකටත් ඔයගලන්ට මේවා අඳින්න වෙනවා, right? A-level paper එකෙත් අහනවා. There are two places like infinity and near. There are two sort of arrangements of placing the light. I can't remember. Physics වලට ඉගෙන ගන්න. Okay. ඒතර ඔක්කොම මේ වගේ ray diagrams අතරයි, yeah, about 6 you have to remember. I had to remember, okay, because ඒ වෙලාවට you don't, sometimes you don't have enough time to apply the logic. Logic එක apply කරලා මෙහෙම නම් මේ wave එක මෙහෙමද යන්නේ. Sometimes you don't have time, right? Because you also have to plan, right? මේ waves මෙතනදී meet වෙන්න ඕනේ. එහෙම there are like it has to happen like that. ඒතකොට ඒ වෙලාවේ මක ආයි race ඇඳන් ඉන්න වෙලාවක් නැහැ. So I'm just advising you. ඔය lesson එක ආවම try to sort of memorize a little bit the ray diagrams, right? Anyway, so today that's enough about microscopes. Today let's quickly finish our chemical basis last part. Okay, chemical basis last part, nucleic acids. Any questions from the things we discussed yet? No, right? So come to nucleic acids, this page, near to it, right? Nucleic acids. So let's read a little bit. Nucleic acids are polymers existing as polynucleotides. Existing කියලා correct කරගන්න. Existing as polynucleotides made up of monomers called nucleotides. Okay. all nucleotides. So what is the most sort of the first fact that you have to know about any biological molecule? If it's a macromolecule and a biopolymer, මේකනේ මුලින්ම අහන්නේ, right? So you have to know if it becomes a macromolecule and a biopolymer. Yes. So your nucleic acids are macromolecules and biopolymers. Yes. Okay. So if it's a polymer, what should be there? There should be a building block, no? Building block එකක් තියෙනවා. What is the monomer එතකොට? Nucleotides. Highlight it. The monomer is nucleotides. The monomer is nucleotides. Okay. Page before. Previous. Then this page. Okay. So then elemental composition is the next important thing you need to know, right? මේවා අහනවා, right? Structure is එකේ what is the elemental composition of this කියලා අහන්න පුළුවන්. So we have our usual four, right? Carbon, hydrogen, oxygen, nitrogen, and additionally we have phosphate, right? Nucleic acids. So nucleic acids are macromolecules and biopolymers. There are two types of nucleic acids: DNA, that is deoxyribonucleic acids, and RNA, that is your ribonucleic acids. Right? We have two types of nucleic acids that we need to know: DNA and RNA. Okay. So here we have a diagram of a nucleic acid. Right? What are the three parts of a nucleic acid? Before that, we need to know what are the parts of its monomer, right? That's what we did when we did proteins also, right? Actual proteins we need to know what is the sort of the structure of the building block, right? That's what we need to know. So what is the monomer? They are called nucleotides, right? A typical nucleotide has three parts, right? A typical nucleotide has three parts. What are the three parts? It has a sugar that has 5 carbons. So what do we call sugars having 5 carbons? Pentose sugar, right? This has a carbon here, 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 and there's an arm like this. There's another carbon here. This is the 5 carbon pentose sugar. This is oxygen, okay? Oxygen. Then connected to this is the first carbon, okay? Here is the first carbon. Connected to that, you have a sort of a complicated group that we call as the nitrogenous base. So this group has our nitrogen, okay, nitrogenous base. There are several kinds of nitrogenous bases as well. So second carbon is here, 3, 4, and 5. Bonded to the fifth carbon, we have a phosphate group, PO4 3 minus. Phosphate group. Okay, this is what a nucleotide looks like, right? So this is a nucleotide. This is what? Pentose sugar. This is, draw this with me, okay? Phosphate group. There's a space, right? There's a space here. Online students, there's a space here, right? So a nucleotide has three components. What are the three components? A pentose sugar, a nitrogenous base, and a phosphate group. What is this sugar actually? This pentose? We actually learned about this sugar. So, yes, it's a 5 carbon sugar. So, when we talked about monosaccharides, we talked about 3 carbon sugars, 4 carbon, 5 carbon, 6 carbon. हम कथा कर रहे हैं trioses, tetroses, pentoses, hexoses, right? So, under pentoses, I gave you some examples, right? Ribose, deoxyribose, ribulose. I gave you 3, right? So, this pentose sugar can be either, okay, completely draw this then. Online people drew this. You have to be able to draw this, okay? In the paper, they can ask you to draw a nucleotide. Connected to the 5th carbon, you have phosphate group. Okay. So, nucleotides can sometimes lose this phosphate group. The phosphate group right? So, they have a free 5th carbon position. So, if you don't have a phosphate group, but you have a nitrogenous base and a pentose sugar, we call it a what? A nucleoside. Nucleoside, not a tide, but a nucleoside. Right? Nucleoside means it has no phosphate group. Okay, that's what it says. So in your tute it says a nucleotide without a phosphate group is called a nucleoside. So we'll learn about adenosine triphosphate. Okay, we are going to learn. So adenosine, guanosine are some examples for nucleosides. Nucleosides. Moving on, let's talk about the pentose sugars, right? Pentose sugars. Pentose sugars are of two types as deoxyribose and ribose. In deoxyribose, one oxygen atom is less than in ribose. Okay, highlight what are the two types of sugars? Deoxyribose and ribose. So, in your pentose sugar, actually here connect to this second and third carbon, you have OH. So, if you have OH, two OHs, this is ribose. OH දෙකක් තියෙනවා නම් this is ribose. If there is only an H, then it is what? One less oxygen ribose. Deoxyribose. So if there is an oxygen here, it's a normal ribose. If there is no oxygen here, it is deoxyribose. That's what they say. Okay. So in the tube, you have a diagram. Yes, this diagram. Online people, can you see? I hope all of you have the tube, right? Think like look closely at this pentose sugar. So tell me, is this pentose sugar, is it ribose or deoxyribose? Online people, put in chat. What is the sugar that is given in the tube? That same diagram is in your resource book as well. It's a deoxyribose, no? It's a deoxyribose. Yes, very good. It's a deoxyribose. Why? Because there's only a hydrogen bonded to the third carbon, no? Third carbon. Sorry, second carbon. Okay? It's a deoxyribose. So, අපි deoxyribose එකෙන් හදන්නේ කවුද? Are we making DNA or RNA from deoxyribose? We are making DNA, no? Deoxyribonucleic acid is DNA. So if we are making a nucleic acid from ribose, it's ribonucleic acid, no? That's a very simple difference, okay? Moving on, so that is all you need to know about the pentose sugar. Nitrogenous bases. There are two major types of nitrogenous bases as purines and pyrimidines. Purines and pyrimidines. Okay, there are two types. So your nitrogenous bases, we have purines, pyrimidines. Okay, there are two classes basically, two types, right? So your purines are larger, pyrimidines are smaller. If you turn to the next page, you have sort of this sort of box that I have given your nitrogenous bases. Can you see here? This box in the next page. Blind people, you can see, right? The two classes of nitrogenous bases, the pyrimidines and the purines. Can you see? The pyrimidines have only one carbon ring, no? They are smaller. But there are three, let's say, types of pyrimidines that you need to know. What are they? Cytosine, thymine and uracil. ඔය විදිහට pyrimidine types තුනක් තියෙනවා. Three varieties of pyrimidines. Then purines, as you can see, they are larger, no? Because why? They have two carbon rings. Can you see? They are larger. Purines. So, what are the two types of purines that you need to know? Adenine and guanine. Clear? Right? So, what are the things you have to remember? You have to know pyrimidines are smaller, and you have to remember the three types of pyrimidines. So, in front of here, no, it's given, no need to write. So, they say purines, there are two types as adenine and guanine. In pyrimidines, there are three types as thymine, uracil and cytosine. So, in any sort of DNA related, let's say, any application of DNA knowledge or whatever, nucleic acid knowledge, we don't write these bigger names, cytosine, guanine, adenine, normally, right? For each nitrogenous base, we give a symbol, right? What are the symbols? So, just what are the two types of purines? Adenine is A, guanine is G. That's it. What are the three types of pyrimidines? Cytosine, thymine, uracil. Those letters, using those letters, we write the nitrogenous bases. Okay. Clear. Shall we move on? Then we need to know, so this is the monomer. When we talk about a monomer, what's the next step? We have to see how the different monomers get together to form the polymer itself. No, that's what we have to know. So we talked about how monosaccharides get together to form polysaccharides. We talked about how amino acids get together to form proteins. So now we are going to talk about how nucleotides get together to form nucleic acids. Okay. Right. Next page, formation of nucleic acids read. Millions of nucleotides join by phosphodiester bond to form polynucleotide chains by condensation between the OH group of the phosphate of one nucleotide and the OH attached to the third carbon of pentose sugar of the other. Right? You need to know. So, what is our new bond that we are learning? Highlight. What is the new bond that we are learning? Phosphodiester bond. Phosphodiester bond, please highlight. That is the new bond that we are learning. So, between what are we having these bonds? OH group of the phosphate of one nucleotide and the OH attached to the third carbon, right? So, if I expand this a little bit, so this is the OH attached to the third carbon, no? That you know, right? So, the phosphate of the below nucleotide, right? So, actually your phosphates have an OH here, an OH here, an OH here, and it is attached to the fifth carbon here, and like this. This is how the below nucleotide looks like. This is the N base, okay? So this is the fifth carbon is here, fourth carbon is here, third is here, second one. Right? So how does the phosphodiester bond form? They say OH of the phosphate and OH of the third carbon. This results in a backbone, right? So there's going to be a condensation reaction. This is again a condensation reaction, right? This is again a condensation reaction. This combination is going to what? Release a water molecule, and there's a new bond, right? Between the third carbon and the phosphate, there's going to be a new bond like this. This is what you can, I think I have mistakenly written it in the tube, right? Yes, phosphodiester bond. Can you see in the green color? Yes, the phosphodiester bond, right? Between, very important, remember, between the OH of the third carbon and the OH of the phosphate. Those are the two groups between which our phosphodiester bond is formed. Okay, got it? Right. Any questions? No. Just remembering that is fine, right? If you like, you can write with your own words, right? Mama ලියලා තිබ්බට කමක් නැහැ. Just draw another arrow and write phosphodiester bond. Right? Then let's read. These bonds result in a backbone with a repeating pattern of sugar phosphate units. Right? Sugar phosphate unit repeating pattern. This is an important statement. එතකොට මොකද්ද? Sugar and phosphate and sugar and phosphate. ඔහොම යන්නේ, right? මෙහෙම යන්නේ. So these people, this kind of this group that is going downwards, these two are sort of independent, or let's say bit separated from this nitrogenous base. Actually, the important person is the nitrogenous basis. That's what gives DNA the variability. For example, so when we learned about amino acids, I hope you remember, right? මෙහෙම carboxyl එකක් ඉන්නවා, hydrogen and NH2 එකක් ඉන්නවා. Who is the important person actually? That the person who changes the amino acid? The R group, no? Yes. ඒ වගේම තමයි මෙතන story එක. This is again going to be a backbone. දැන් අපිට amino acids වලත් මේ set එක කරේ මොකද කිව්වේ? The backbone, right? So in nucleic acids also, this phosphate sugar and the pentose sugar and the phosphate are going to be form the backbone, right? Nitrogenous bases tamai important, right? They are sort of outside the backbone, right? Can you see in this diagram the pentose sugar and the phosphate groups are sort of in a blue box. That blue box is indicating what? On blue background they have given also on the top. That is the sugar phosphate backbone. Okay, sugar phosphate backbone. So these bonds result in a backbone with a repeating pattern of sugar phosphate units, right? ඒක ටිකක් මෙහෙම යන්නේ මං කිය. Sugar phosphate sugar phosphate sugar phosphate එහෙම යන්නේ, right? One after the other. Nucleic acids are linear polymers of nucleotides, right? There are two kinds of nucleic acids depending on the type of the sugar molecule involved. If the sugar is deoxyribose, then the nucleic acid is DNA. If the pentose sugar is ribose, then the nucleic acid is RNA. Highlight those. Highlight those. Any connection issue? Online people, you can clearly see me and hear me, right? Just give me a feedback. Please send to the main chat, okay? If there's a chance that I'll miss your message if you send a private message, okay? Clear? Simple things, right? I don't want to go into massive detail. I don't want even to draw this formation in much detail because you don't have to draw it. So I don't have to draw it, right? I will draw only in the board things that you have to draw by yourself in the paper, right? There's no use wasting time. Okay. Okay. So formation of nucleic acids clear. Simple things. What do you need to know? There's a new bond called phosphodiester bond, right? Forming between the OH of the third carbon. So this is the phosphodiester bond. Third carbon of the pentose sugar and OH group of the phosphate group that is going to connect the sugars to the phosphate, leading to a sugar phosphate backbone, right? Sugar phosphate backbone. And if the sugar is ribose, it's going to produce ribonucleic acids. If the sugar is deoxyribose, it's going to produce deoxyribonucleic acids. Simple. Moving on, we are going to talk about DNA first, right? DNA. Structure of the DNA molecule, Watson and Crick model. Watson and Crick model, right? So can you silently read this while I set up the projector? Can you silently read this paragraph? Watson and Crick model. So this DNA, like the discovery of the structure of DNA was an important, let's say, a milestone in biology, right? Because in like we'll talk about why DNA is so important for life. Okay, so because of this discovery of this DNA double helix, right? It lead to sort of a jump in the field of genetic studies, molecular studies, everything related to DNA, right? So actually this double helix was actually discovered by a technique called X-ray crystallography, right? X-ray crystallography, you will, depending on where you go for chemistry, they will touch a little bit when you are doing structure and bonding. Structure and bonding start kara. Second lesson, start. So there you have to draw these Lewis structures, everything, right? So actually we can see with our own eyes the structures of molecules using a technique called X-ray crystallography. Okay, I don't exactly know how it's done, but there's a technique, right? Using that technique, you can actually see how a molecule looks like. So the person called Rosalind Franklin, a lady, actually did the X-ray crystallography on the DNA molecule. So she actually sort of, we can say, found out the structure, but Watson and Crick actually described it. Okay, Watson and Crick actually described it. So the credit actually goes to Watson and Crick for the actual identification of this. Sorry, actual the presenting of this structure, the credit goes to Watson and Crick. But who actually discovered it was Lynn Franklin. So this is what we learned, right? If I zoom in, yes, can you see? If you look from the top, This blue things are what? The pentose sugars, right? Pentose sugars. This purple color, these now blue color ones, those are the what? Phosphate groups, right? Can you see? As I told you, they are arranged one after the other, right? Phosphate, sugar, or sugar, phosphate, like that. Can you see? So this, can you see? It becomes this blue color strip, no? So our sugar phosphate groups will form what we call as the backbone, right? Backbone of DNA. So if we zoom out our DNA, this is a classic appearance. So you have seen this so many times, right? We have seen DNA, seen, and in labs we have so many models, and everywhere we go, you have the structure of DNA. So this looks like a spiral staircase, no? Right? So the, what you say, the handrails attata deka thamai mokadda? The backbone, sugar phosphate backbone that twist around. And the stairs padi monada? In the center you have your what? Nitrogenous bases, right? Can you see the colorful nitrogenous bases? So now you know what are the two types of nitrogenous bases. What are the two types? What are the two types of nitrogenous bases? There are smaller what? Pyrimidines. Small people are the pyrimidines, no? Only one carbon ring. Can you see? Only one carbon ring. Yes. Here. So those are, they must be what? Pyrimidines, no? Small ones. Only one carbon ring. Two carbon rings. What are those then? They are purines, right? We don't know what kind of purine, but according to the structure, you can see. You can say they are definitely a purine, right? Two carbon rings. Get it? So this is the double helix structure. So there's another view. This is what I showed you, right? Yeah. This is the double helix. We'll learn about this, how the DNA is actually tightly packaged inside cells, right? Tightly packaged inside cells. It's like this, right? If you take a cell, the DNA is nicely packed into the cell in an organized manner, right? So before that, I want to tell something that is called the base pair rule. Okay, the base pair rule. I will show that using this. The base pair rule. There's a sort of a universally accepted way that these purines and pyrimidines bind. So the first thing you need to know is always, first point, always a purine binds to a pyrimidine. Binds to a pyrimidine. That's the first rule. Okay, purine join වෙන්නෙත් නැහැ, pyrimidine join වෙන්නෙත් නැහැ. Okay, and second thing, they join according to a rule. Next thing, they join by hydrogen bonds. Okay, they join by hydrogen bonds. And this, so what are the purines that you know? Adenine and guanine, no? Right? Adenine always binds with cytosine. Right? Adenine always binds with, sorry, thymine, okay? AT, okay? Adenine always binds with thymine, okay? By two hydrogen bonds. Adenine binds with thymine by two hydrogen bonds. Guanine binds with cytosine by three hydrogen bonds. This is the base pair rule. ATGC. Okay? ATGC. You can see that here in this diagram. So can you see? If we zoom in here, okay, this one is adenine loop. Can you see? It's adenine. So who is supposed to be binding with adenine? So adenine is a purine, no? It has to bind with a pyrimidine. Who is the pyrimidine of adenine? Thymine, no? Can you see? It's thymine. how many hydrogen bonds can you see? There's one here, there's one here. 2 hydrogen bonds. Then this one. They say this one is guanine. Guanine, according to the base pair rule, has to bind with 3 hydrogen bonds with what? Cytosine. Yes. So can you see here? 3 hydrogen bonds. Online people, you can clearly see the board, the projected screen, right? Yeah. So come to the tute again. Come to the tute again. Structure of the DNA molecule, the Watson and Crick model. DNA molecules have 2 antiparallel polynucleotide chains that spiral around an imaginary axis, forming a double helix. Right? අපි මෙහෙම spiral පඩිපෙළක් ගත්තත් නිකන් මැද either නිකන් මැදින් පොල්ලක් වගේ එකක් යනවනේ. Spiral staircase එකක් නේද එළියක. Yeah. Right? It spirals around සාමාන්‍යයෙන් spiral staircase එකකට මැදින් ඔහම ලොකු කණුවක් යනවානේ. ඒක වටේටනේ weld කරලා තින්නේ if it's a metal staircase. But DNA වල මැදින් එහෙම කණුවක් නැහැ. Right? It's only imaginary. It spirals around imaginary other කණුවක්. Right? So anyway, it looks like a helix. Okay. So I'll tell you what this antiparallel is, right? But for now, highlight 2 antiparallel polynucleotide chains that spiral around an imaginary axis, forming a double helix. This is important, right? The two sugar phosphate backbones run in opposite directions. I'll come back to there. Okay. Antiparallel katawa I need to explain, so I'll come back there. The sugar phosphate backbones are on the outside of the helix, and the nitrogenous bases are inside the helix. Can you see? So these are the sugar phosphate backbones on either side, no? The 2 backbones, and these are the stairs, no? Padipela wage mem stairs tika. So these are the nitrogenous bases that are inside the helix. Okay, inside the backbone. The 2 strands are held together by hydrogen bonds between the paired nitrogenous bases. So can you see hydrogen bonds දිගටම උඩට වෙනකන් තියෙනවා. That's what keeps the 2 nucleotide chains connected to each other. මොනවගේද connect වෙලා තියෙන්නේ? Hydrogen bonds වලින්. Then base pair rule. Always a purine base pairs with a specific pyrimidine base. Highlight. Right? A purine always binds to a pyrimidine. Then under that, write down this ATGC. ATGC. Adenine binds it. I mean by how many? Very important. How many hydrogen bonds? 2 hydrogen bonds. Guanine with cytosine with 3 hydrogen bonds. Right? Okay. That is the base pair rule. එතන ඉඳ තිබ්බනේ. එතන තිබ්බ තැන ලියාගත්තනේ. Base pair rule කියන එකට there's some space here. Right? Okay. So what is this anti-parallel? Right? What is this anti-parallel? If you take a look, right here, it's very difficult to zoom. So these are carbon atoms, no? Right? So the phosphate always binds to the 5th carbon, no? The phosphate always binds to the 5th carbon. It's a bit difficult to show with this anti-parallel. Hold on. I think there was a nice diagram in the Campbell. So this is the same diagram I have in the tute, right? So I'll just explain using this. Okay. So this is the first carbon, second carbon, third carbon, right? So third carbon is sort of now exposed on this side, right? This is called, so if the third carbon is the sort of the end of the chain, this is called the 3' end. 3' end. The third carbon OH is sort of now hanging out. It's sort of the end. On this side, what is the end? Right? It's the fifth carbon, no? That is attached to the phosphate. So the top end is called the 3' end. Can you see? This is called the 3' end. So this chain runs, we say the bonds are formed from 5' to 3' downwards. Right? Always these phosphodiester bonds, there's one here, there's one here, there's one here. These phosphodiester bonds are always formed from 5' to 3'. There is a rule. The thing is, so you know, here you have to have, if we assume this is what? If you assume this is guanine, there has to be a what? A cytosine here. Yes, there has to be a cytosine here. This cytosine is bonded to a pentose sugar, right? And it's bonded to a phosphate like this, you know. The thing is, this chain is actually oriented upside down, right? Yeah, ඉන්නේ මේ අනිත් පැත්ත. මෙහෙම නෙවෙයි ඉන්නේ, right? The other polynucleotide chain that is sort of supposed to be bonded to this one is sort of upside down like this. Upside down like this. So the phosphate I'll draw as P. It's like this, right? This is what now? Again, 1, 2, sorry, 1. This is 1, no? Here is 1, 2, 3. This is the 3 OH, no? So this is the 3' end of the other chain that is going to go down. Yes? And here also you have this. Here, it's supposed to be a purine, no? Methane, right? Because this is a pyrimidine, no? Methane, purine connect. So here is the 5' end. So this chain runs in this direction. Also 5' to 3'. But the two chains run in opposite directions. Can you see? This one runs downwards from the 5' to 3'. මෙයාගේ 5' එක තියෙන්නේ this side and the 3' on this side. So it runs on the other direction. Right? No need to worry about it that much. Just remember the two chains are not running in the same direction but opposite. Right? Anti-parallel. Right? Is it clear? No need to be able to draw this. Okay? But you need to know the fact that the two chains that are connected by the hydrogen bonds are running in opposite directions. What is the direction? 5' to 3' always. Right? එතකොට මෙයා 5' to 3' downwards නම් මෙයා 5' to 3' upwards. That is basically what you mean by anti-parallel. Okay? So after the base pair rule, we have a small paragraph Hence, the strands of DNA are said to be complementary to each other. The pairs are known as complementary base pairs. And in this original double helical structure, one complete turn consists of 10 base pairs. Right? One turn has 10 base pairs. It's like this. Can you see? So this is one turn, no? So this is one turn, no? In between here and here, one turn. 1, 2, 3, 4. You can only see 4 here, right? In this diagram, this one also has 4. So one turn is supposed to have 10. So 5 on either side. In the software, how many do we have? One turn has 1, 2, 3, 4, 5. It has 5 again. Can you see? So 5 on one side means altogether 10, no? So if you look, start from here, 1, 2, 2, 3, 4, 5 again. Can you see? Between two turns, 5 base pairs on one side. So that means altogether 10 on the two sides. Yes. Okay. What else do I need to show you? Anything interesting? Not really. These animations not that important at this stage. We'll talk about this when we are doing the nucleus, right? There's no audio. This mRNA I will show you because we are going to learn about this mRNA. So this is the DNA, right? Can you see the double helix? Audio. mRNA we'll talk within today, right? This is mRNA, okay? I'll show this animation again when I have taught you different kinds of RNA as well. Okay, right. So moving on, that is all you need to know about DNA. Oh no, finally we have to know the functions of DNA. Right? Functions of DNA. Online people, any questions? One turn consists of 10 base pairs. Yes, 5 on one side, right? That's the logic. 5 on one side. So altogether 10, no? Yeah. 10 on one side. So single turn means like one twist, right? 10 base pairs means on both sides. So on one side there's 5. That's the logic, I think. Okay. I think in the diagram given in your resource, you actually have nicely 5. There's one hidden at the top, right? You have 5. So on both sides you have 10. Moving on to functions of DNA. Functions of DNA. So why are we so concerned about the structure and how it's arranged? All this because the DNA plays a massive role in deciding everything that we do, right? In everything our cells do. So in sense, it decides what we do, right? Because we are a collection of trillions of cells. We within a collection of cells, no, a ball of cells, right? So each individual cell is controlled by the DNA inside that cell. So we are in turn controlled by our this molecule, this DNA, right? So the most important function is store and transmit genetic information from one generation to the next generation. So we'll talk about when we're doing cell division, right? Inside our cells, we have a nucleus, right? Inside the nucleus, we have the DNA. So when one cell divides into another cell, that DNA will also be transferred to the new cell, right? So the DNA is sort of like the right? From one generation to the next, the DNA will be given. So by looking at the DNA, the new cell will decide how am I going to work, how am I going to behave, right? That is how a new cell behaves like the old cell, right? Because it's inheriting, it's receiving DNA from the previous generation. Okay. Then store the genetic information for protein synthesis. Okay. So I'll show you this process of how we can make proteins from DNA. It's a nice process, right? So it's like this. So you are going to make a cake, right? You are going to borrow a cookery book from the library, right? How to make a cake. Cake එක හදන්නේ කොහොමද? cake එක කියලා imagine කරන protein එක, right? So මේ protein එක හදන්න, ඒ කියන්නේ cake එක හදන්න ඔයා පොත ගන්න යනවා library. So library කියන්නේ මොකද්ද? Knowledge is stored, right? So in a cell, the library is the nucleus, right? Where all the books are nicely stored there, right? The books are what? DNA, right? So you are going to make a specific type of a chocolate cake or something. So what you go to the nucleus and you go through the library and find out, yes, this is the recipe for making chocolate cake. But the problem is, library you are not allowed to borrow books, right? You need to note down or make a photocopy of this recipe inside the nucleus, right? You have to like copy the recipe. Cake එක හදන හැටි copy එකක් ගහගෙන හරි note එකක් ගහගෙන හරි තමයි එළියට යන්නේ. So ඒ recipe එක අරගෙන ආ cell එකෙන් එළියට එනවා. sorry nucleus එකෙන් එළියට ඇවිල්ලා එළිය ඉඳන් තමයි cake එක හදන්නේ. මේ විදිහට තමයි සාමාන්‍යයෙන් අපේ protein හදන process එක වෙන්නේ. protein හදන හැටි තියෙන nucleus එකේ protein හදන්නේ එළිය outside the cell, right? This is a nice process that is called the protein synthesis. Okay, I'll explain it using the animation as well. So මේ ඔක්කොම recipes ටික protein හදන recipe ටික ඔක්කොම තියෙන්නේ stored as DNA, right? DNA විදිහට තමයි මේ ඔක්කොම information of how to make proteins is stored as DNA. Okay, highlight those two. Very commonly asked what are the functions of DNA. Okay, any questions online, people? No, right? Okay. So shall we move on to RNA, right? So this RNA is very commonly associated with DNA as well. DNA එක very closely වැඩ කරනවා RNA, right? There are several differences of RNA compared to DNA that you need to clearly know. Okay, let's just read structure of RNA. This is normally a single stranded nucleic acid composed of the ribonucleotides. That's the first difference, right? RNA වලින් what pentose sugar? Ribose, not deoxyribose, right? Ribonucleotides containing the bases uracil, cytosine, guanine, and adenine. Now, right? So who is missing in the RNA? There is no thymine. There is no thymine, right? Instead of thymine, you have the other pyrimidine. What is the other type of pyrimidine that we learned? Uracil, right? That's another big difference in RNA. There is no thymine in RNA. Instead of thymine, you have uracil. You write thymine like this, okay? Thymine. Thiamine කියලා කෙනෙක් ඉන්නවා. That is vitamin B3 එතකොට. It's a vitamin, okay? Not thiamine, thymine, okay? So there is no thymine in RNA, there's only uracil, okay? Complementary base pairing between two RNA molecules or within the same molecule may occur in some. Usually, RNA doesn't have that sort of a helix structure or two chains don't join like in DNA. RNA වල එහෙම නැහැ. Alright? If there is a complementary binding like this, it always occur within the same RNA, right? එකම RNA molecule එක ඇතුළේ විතරයි වෙන්නේ. So when you are making the DNA double helix, two completely separate chains got together to form the double helix by making these complementary bonds, hydrogen bonds. But when we take RNA, එයා මෙහෙම complementary bonds හැදුවා, ඒවා හදන්නේ එයාගේ ඇතුළෙමයි, within the molecule. But if that happens, there is another එයාටම ආවේනික base pair rule එකක් තියෙනවා. That is again same, very same. Adenine binds with two hydrogen bonds to uracil. That's the only difference. Guanine binds to cytosine with three hydrogen bonds. This is the only difference. Okay. Complementary base pairing facilitates a three-dimensional shape essential for their functioning. Adenine binds with uracil by two hydrogen bonds and guanine binds with cytosine with three hydrogen bonds. Okay. So if you want, you can quickly write this down. The new base pair rule for RNA. Okay. Base pair rule for RNA. Right. So there is a variety of RNAs. Like compared to DNA, we only have the DNA double helix, right? There's only one kind of DNA. But RNA, we have three important types of RNA that we need to study. In several places of the syllabus, we have to again and again come across these kinds of RNA. Especially, we are going to study them now, and then we are going to study them when we are doing molecular biology, right? Lesson number 8, right? Very, very important there. We have to deal a lot with DNA as well as a lot with RNA as well. Okay. So write down, if there's 1, 2, 3 numbers, there are three types of RNA present in cells. Three types. What are the three types? Write down. First one, messenger RNA, or within brackets, mRNA. Messenger RNA, mRNA. Second one, transfer RNA or tRNA. tRNA. The T is and M is simple, RNA is capital. Okay. And finally, ribosomal RNA or rRNA. Okay, these are the three types of RNA that we have in our syllabus. Each performs a unique role, right? එයාටම ආවේනික roles තියෙනවා මේ RNA වලට. Okay, so let's talk first about messenger RNA. Let's read then I'll explain. Okay, messenger RNA is a linear molecule and is the least abundant type of RNA. So if you take a cell and if you collect all the RNAs present in that cell, අඩුවෙන් ඉන්නේ mRNA, messenger RNA. Okay. There are two functions of the mRNA. Copies the genetic information stored in DNA molecule as a sequence of nitrogenous bases. Transport the genetic information from the nucleoplasm to the site of protein synthesis, ribosome to nucleopores. Complicated thing, right? So let's talk about mRNA. I think better just let's read all types of RNA, then I can explain it as a complete story, okay? Move on to the second type of RNA, tRNA. Smallest RNA molecule, it's linear but forms a 3-loop structure as shown in the diagram. Can you see the 3 loops? Function, transportation of amino acids to the site of protein synthesis. Okay. Then finally, ribosomal RNA, it is the most abundant type of RNA. It has a complex irregular structure, provide the site where polypeptide chains are assembled. Right? Right. Okay. So now we are going to make proteins, right? With the help of these different kinds of RNAs. We are going to make proteins. So imagine it's a cake. Okay, cake කතාවනේ තියෙන්නේ. Cake එකක් හදන්න අපිට මොනවද ඕනේ? Kitchen එකක්, right? Oven එකක්. ඔය kitchen එකක් එපා බඩු ටික තියෙන. So where do we make proteins in a cell? There's a special organelle, no? Called ribosomes. Ribosomes කියලා චුරි organelles. No, they are not organelles. We call them subcellular structures. Ribosome එකේ තමයි proteins හදන්නේ. That's the kitchen, right? So our ribosome is actually composed of our rRNA mainly, right? Ribosomal RNA contributes to form the ribosome itself, right? Ribosome එක හැදිලා තියෙන්නේ mainly rRNA වලින්. There are some other proteins involved as well, right? So can you see? It provides the site where polypeptide chains are assembled. So ribosomal RNA form ribosomes. Okay, so that's the kitchen. Then in the kitchen we have an assistant. නිකන් අර කුස්සිය අපිට assistant කෙනෙක් ඉන්නවා. අපිට මේක ඕනේ කිව්වහම මේක ගැල්ලා දෙන්න, මේක ඕනේ කිව්වහම මේක ගැල්ලා දෙන්න, right? We have an assistant to make this protein, right? Those are our transfer RNAs. Those are our assistants. Okay? එහෙම ඔළුවේ තියාගන්න. ඊළඟට messenger RNA කියලා කියන්නේ the recipe book that we bring from the library, right? That recipe that has how to make the cake is your mRNA, right? Because why? The library doesn't allow you to take the recipe book outside. Nucleus එකෙන් එළියට ගෙනියන්න දෙන්නේ නැහැ DNA. So what do you have to do? You have to either make a photocopy of the recipe and then you have to bring it out of the library nucleus. That's what you need to do. It's not easy. So in this process of protein synthesis, we have 2 steps. Okay, these are not out of syllabus, right? You have to learn them within your 2.5 years. So why not learn a little bit? You might forget, but subconscious එකේ හරි කොහේ හරි හැංගිලා තියෙන ඉගෙන ගත්තා කියලා, right? So there are 3 steps. So DNA is your recipe, right? Library එකේ තියෙන cake එක හදන හැටි තියෙන්නේ මේක DNA වල, right? So DNA ටික එළියට ගෙනියන්න දෙන්නේ නැහැ. DNA is located where? Inside the nucleus. So this is the nucleus. You know this, no? Cell එකේ there's a nucleus and inside that you have DNA. Here is your ribosome. Ribosome is looks like this. It has a large subunit. It has a large part and a small part. This is what your ribosome. Online people, you are following, yes? This is your ribosome. Okay. So මෙයාගේ assistants ලා විදියට near the ribosome you have your transfer RNAs. I will draw you the transfer RNAs bit like this, right? These are tRNAs. These are assistants at the kitchen. Ribosome, right? Then මොකද කරන්නේ? So ribosome is mainly composed of what? rRNA and some proteins. Okay. So So now we can see in this diagram the two of the three types of RNA, you know. So what do we have to do? We have to bring the information present in the nucleus in the DNA. We have to bring it to the ribosome. There's a middleman to do that. How the middleman? There is a messenger RNA, mRNA. That is a photocopy. So by looking at your DNA, we are going to make an mRNA messenger. And your nucleus actually has, this nucleus is covered by a membrane. And that membrane has this sort of gates, openings there. Okay, it has these sort of gates like this. Right? Several tangle, right? These are called what? These are called nucleopores. So the inside you can actually call it the nucleoplasm. Inside this nucleus is called the nucleoplasm. You don't. It's not actually very accurate, right? So, sorry. අපි දැන් මේ තියෙන පොත recipe book එක දිහා බලාගෙන we had a photocopy of the recipe. That is your MR. So we are bringing the photocopy out of the library to where? To the kitchen. The messenger RNA brings the message in the DNA to the ribosome. This is the purpose of mRNA. So this step, this process of copying, photocopying the information in DNA to mRNA is called transcription. Okay, transcription. You can see it in the diagram I have given in the tute, right? Can you see? From DNA to mRNA, the process of photocopying a DNA to mRNA is called what? Transcription. And here, at the ribosome, we are going to produce a protein. That is the product. A protein is like this. What is this? A chain of amino acids. What do you call the chain of amino acids? Yes, last week, week. These are amino acids. Joined by what bond? Amino acids are joined by what bond? Peptide bonds. Yes, amino acids are joined by peptide bonds. You are following, right? Yes. So this is what? A polypeptide chain. This is our cake, the product. So the process of converting the information into a protein or an amino acid chain is called translation, right? This process is called translation. Two processes need to happen to produce a protein from DNA. Okay? You have to make mRNA by looking at DNA. That mRNA will be converted to a protein in the ribosome. Clear? So that is what I have given in this orange color box. This is a summary of how protein synthesis happens. Right? Transcription and translation. These are sort of bit deep this early, but you will have to actually write an essay, write essays from transcription and translation, right? The idea about transcription and translation. Right. Podda confused right? So this one, focus on this person, our polypeptide chain. This is our final product, right? Polypeptide chain protein. Composed of what? Amino acids, right? Amino acids. How many amino acids are involved in making proteins? How many types of amino acids are needed to make a protein? 20. 20 types of amino acids we can use, right? So the interesting thing is this tRNA brings the amino acids to the ribosome, right? Then what are we going to make? We are going to make proteins, right? So imagine this is the cake. කේක් හදන්න මොනවද ඕනේ? පිටි ඕනේ, සීනි ඕනේ, බටර් ඕනේ. ඔය ටික එක එක ඒවා ගෑලා දෙන්න එපා. So those raw materials are what? The amino acids. Each amino acid, to bring each amino acid, there are separate assistant. Assistant ලා 20ක් 20 types of assistance. For example, so if the cake, butter ගේන්න වෙනම එක්කෙනෙක්. Sugar ගේන්න වෙනම එක්කෙනෙක්. Eggs ගේන්න වෙනම එක්කෙනෙක්. Okay? For each ingredient, there are some separate assistant. So for each amino acid, you have a separate tRNA. Okay. That's something you need to know. Right? එතකොට මේ there's a special, so one kind of tRNA brings only one kind of amino acid. Right? දැන් ඔයගොල්ලෝ දන්න amino acid types මොනවද? There's one, you know, glycine, right? You know, glycine, simplest amino acids, you know. So there's a dedicated tRNA that is only bringing glycine. එයා දන්නේ glycine එක ගේන්න විතරයි කඩේට ගිහින්. Right? So in your cell there is a amino acid pool එකක් තියෙනවා. නිකන් කඩේ වගේ amino acids ටිකක් තියෙනවා. Right? So the tRNA of glycine only knows to how to bring glycine. He doesn't know how to bring any other amino acid. There's a story like that. Okay? I think that is enough for at this stage. Right? So tRNA, what is the function? Highlight. Yeah, very good. 20 amino acids. Transportation of amino acids to the site of protein synthesis. What is the site of protein synthesis? Online people? The site of protein synthesis? Your ribosomes. Ribosomes are mainly composed of what? Ribosomal RNA and proteins. If you like, you can take this down. It's basically the same thing that is given here, right? Same thing that is given here. If you like a diagram of your own, you can quickly take this down. So I'll show you a little bit about this, how these different kinds of RNA involved in protein synthesis. Okay, it's a much more complicated process, but just have a sort of a issue, slight idea in your heads. නැත්තම් transcription translation කියලා මොකක්ද එකපාරටම molecular වලදී හම්බුනහම don't be that confused. Okay. Okay, මේක ලියගත්ත නේද? It's not necessary, but just sometimes it's good to have one of your own diagrams, right? Okay, so these are the two steps, right? Transcription and translation. Let's see how they happen inside a cell, right? So I already showed you the transcription one. There's no sound, so I'll be the sound, right? So we are going to go inside the cell. Now this is the nucleus. Okay, online people, you can see the screen, right? Any issues? No, right? So this is the nucleus, right? Inside the cell. These green color. Those are nucleopores, gates into the library. Library ke dwarwal, right? So nucleopores. Then we are going to go inside the nucleus through a pore. Inside the nucleus we have DNA. So this is our mRNA. That is what our photocopy, right? So we are going to make an mRNA by looking at our DNA. So that's we are making mRNA. Can you see? mRNA is a linear molecule, right? So we are making a photocopy of the cake recipe of how to make the cake, how to make the protein. So that is the mRNA being made, right? So this is like opening a book and photocopying, right? So the DNA is sort of unraveled, and we look at the page we need, and then we take a photocopy. And can you see the mRNA is coming out of the nucleus through the nucleopores? So that is transcription, no? DNA के mRNA का खादने. Translation on. So the mRNA now comes out. It goes to where? To the kitchen. That is what? This is a ribosome, right? Ribosome is the kitchen. This is one half of the ribosome, and this is a tRNA. Okay, it's the assistant. You will see this anticodon codon stuff not necessary at this moment. This is the tRNA. Can you see one end of the tRNA? He is holding an amino acid, right? To make the cake, on butter ගෙනාවා. He has brought an amino acid, right? That is the function of the tRNA. He brings the amino acid. Then that is the big part of the ribosome, right? Now it's complete. On the second second tRNA has come. He has brought the second amino acid. So now the two amino acids are going to join. By what? By peptide bond formation. And the first assistant now goes out, right? He'll go out. Yeah. He is not necessary. The tRNAs are bringing amino acids continuously. And can you see the chain is growing? The protein is being made, right? So the tRNAs are coming in a nice order, and the protein chain is growing. Okay? And at the end, this is what? This is now a primary structure, right? Primary structure of proteins. This will undergo some different changes into secondary structure, tertiary structure. We'll get a nice cake rice in දාලා උඩින් එක මල් ගහලා අර මේක කළා වගේ we get a nice final product. Okay, this is how all these RNAs get together to do protein synthesis. Any questions? No. So let's quickly talk about the differences between RNA and DNA, and then you can have a break. After the break, we'll be doing the questions at the end, right? There's a few questions that we need to do from the nucleic acids. Okay, so these differences between DNA and RNA is a very important comparison that you need to be able to do. Okay, we have to be able to compare DNA and RNA. What are the criteria? DNA, RNA. What are the criteria? DNA is, yes. How many strands does DNA have? Usually double strand. DNA double stranded. Usually RNA single strand. Right? Single strand. what are the nitrogenous bases that you can see in DNA? Adenine, thymine, guanine, cytosine. In RNA, what do you have? Adenine, uracil, guanine, cytosine. Okay, that's the second difference. Third one, DNA has what pentose sugar? Deoxyribose. RNA has what? Ribose. Okay. Better if you can sort of draw a slide chart like this quickly. What will happen to the rRNA after translation is done? The kitchen stays, right? Kitchen, so it's like this. As you saw in that animation, the two parts of the ribosome comes together to form the protein. When the protein is made, I mean, it's completed, it sort of gets separated. But they sort of float around until they are needed to make another protein. Okay, so the ribosome actually stays. I mean, all of these structures will be recycled after some time. But a protein it does not get destroyed, right? The ribosome stays for some time. It's going to make a lot of proteins. Then it's going to be destroyed definitely. Yes, after some time. Okay. So always RNAs are single stranded. Okay. RNAs are single stranded, but does that mean there cannot be complementary binding? Right? Does that mean there cannot be complementary binding? No. If you take a molecule like tRNA, for example, this is still the same RNA, no? Sorry, same RNA, no? Same chain. But within it, right? Within it, there can be complementary binding like this. It's the same chain, but within itself, there can be some complementary binding, but never two strands, right? Molecules දෙකක් එකතු වෙන්නේ නැහැ. There's only one strand, but within it there can be complementary binding. This is a tRNA, no? Okay, don't be confused about that, right? Clear about the differences between RNA and DNA. Okay. So, shall we talk about other nucleotides? Or shall we do a question? So actually, we have to write a essay, right? That's what we need to do. There's not much questions from DNA and RNA, like literally from DNA and RNA. So I think let's take a break now, and then we'll come back and do the nucleotides other than those found in nucleic acids. Then we can quickly do the questions from nucleic acids, and then we can move on to the cellular basis today itself. Okay, the other plasma membrane what you can try. Okay, take a break, 15 minutes. Online people, 15 minutes. It's now 10:25. Come back at 10:40. No, 10:40. Okay, take a small break. If you have any questions, you can ask me now. So this base pair story, right? What does your resource say? One complete turn contains 10 base pairs, right? 10 base pairs. So you're saying 20 bases are there. You can take break. Go, go. Mm,
Yeah, yeah.
So you're right. I'll talk about this number of base pairs again. Yeah, I just looked at some of my notes. Yes. Yeah. Okay. Okay. Come back at 10:40. Okay. Can you hear me? Online people, can you hear me? Okay, join again. Let's start. Okay. Right. The thumbs up is going like this. Okay. Zoom එකේ අලුත් feature එකක් තියෙන thumbs up එක දැම්ම මෙහෙම යනවා thumb. Right, okay. So let's quickly get over with the nucleic acids. Yes. Then you get a thumbs up. Okay. Right. So we talked about the differences between DNA and RNA. So how many nucleotides did we actually talk about? Ribonucleotide and deoxyribonucleotide, right? So those are the types of nucleotides that are involved in forming nucleic acids, right? ඒ කියන්නේ polymers වල monomers විදියට ඉන්න අය විතරයි අපි study කරේ. Those are the deoxyribonucleotides and nucleotides. But deoxyribonucleotides and ribonucleotides. But there are other nucleotides that are not involved in forming nucleic acids, right? They don't necessarily make these kind of long polymers, but they are still what? Nucleotides, right? So we are going to learn about 4 of those nucleotides, okay? So first one is ATP. ATP is called the universal energy carrier, right? So this is kind of interesting because we use a currency, right? We use a currency, you know, like I mean, within Sri Lanka, it's the rupee, right? We can give rupees and get a service back, right? So to do something inside the cell, we need something. So we need energy, no? To do whatever, any kind of work inside the cell, we need some kind of energy. But this energy cannot be different forms, right? This energy that is used inside the cell must be the same kind everywhere in the cell. And also it has to be same everywhere in the same organism also, right? නිකන් එක රටක් ඇතුලේ currencies ගොඩක් use කරන්නේ නැහැ අපි. Within the same country we use the same rupee currency, otherwise it would be very confusing, right? එක්කෙනෙක් ඇවිල්ලා dollars දෙනවා, එක්කෙනෙක් ඇවිල්ලා dinars දෙනවා. Then it will be, there's like, what is the conversion rate, how do I use it? ඒ වගේ there's going to be confusion. So within the cell as well as within the same organism, we use the same molecule as the energy form, right? That molecule is called adenosine triphosphate, ATP. Okay, adenosine triphosphate. Read, it is the universal energy carrier. So usually in all animal cells, right? Usually in all animal and sort of eukaryotic cells, we have ATP as our energy, high energy molecule. So let's draw an ATP molecule. You need to be able to draw this. We'll do this when you're doing metabolism again. ATP. It's a very high energy molecule. It's like this. There's the ribose. Pentose sugar is the ribose. There's an N base, that is adenine. Okay, adenine. And connected to this fifth carbon, you have not only one, not two, you have 3 phosphate groups. This is ATP. This is a ribose, okay? This is ribose. This is ATP. Adenosine triphosphate. Adenosine triphosphate. So there's a way of using this energy, right? So this energy is actually concentrated in these 2 bonds, right? When you break this bond, you release energy, okay? So usually we don't break both bonds, we only break this bond, okay? only the terminal phosphate bond we break, right? So this ATP we can use to give energy. It will become adenosine diphosphate plus a phosphate group plus what do you get? Energy. This is the reaction, right? Or supermarket you can give ATP and you can buy something, no? Like that. If you go to the, let's say, within the cell, tell me something that is working, right? Like if you give, so inside the cell you have transport proteins, no? So if you give ATP to that sort of transport protein, it will start to move like that using the energy of this ATP molecule, right? This ATP is what we use to give energy. And you will see that when we are studying about respiration, photosynthesis, things like that, our ultimate goal is to produce ATP. We have ATP, right? ATP. Right? Drew this, drew the ATP molecule, and also write down this reaction, how energy is released. Okay. Clear about ATP. Then moving on to the second nucleotide, that is NAD plus. NAD plus. Can you see? What is NAD plus? The long name is nicotinamide adenine dinucleotide. If you want, you can write, right? No need to know the long name. It's nicotinamide, okay? Nicotinamide adenine dinucleotide. No need to know the structure, okay? NAD plus. Okay. So what are the functions of NAD plus that they have given? Act as a coenzyme, act as an electron carrier, function as an oxidizing agent during respiration. That's Mulima, what is this coenzyme? Right, we'll talk about this when you are doing enzymes, okay? Your coenzymes are coming under a group of things that we call as cofactors, okay? Cofactors, right? So you know what an enzyme is, right? You know what an enzyme is, right? An enzyme sort of pushes a reaction to happen, right? So imagine you want to break down a disaccharide into two monosaccharides, right? You can put an acid and heat it, no? That's what we did, right? The last week we converted sucrose into glucose and fructose, no? That's what we did. Acid එකක් දාලා heat කරා. Right? But in our body, can we heat it to until it boils? අපේ බඩේ, if you eat sugar, sucrose ටිකක් කෑවොත්, do we add acid and boil it? No. We add an acid, yes. But do we boil it? No. We have to find some other way in living systems to do reactions, no? So heating is giving it some energy, no? Right? But in living systems, we cannot give that much high heat. Destroy if we try to heat. So in living systems, we have found, I mean, organisms have found this kind of enzymes that they act as catalysts. Have you heard catalysts? Catalyst, they can like push a reaction to happen, right? At low energy levels, they can promote the likelihood of a reaction happening, right? We make a mechanisms igana gan, right? So in chemistry, you are going to learn anyway about all kinds of catalysts. In bio, before that, you have to learn about how catalysts promote reactions. Activation energy, the catalyst, what the catalyst does is it reduces the activation energy. So at low temperatures, reactions can happen, right? So in our stomach, to break down, imagine sucrose to the glucose and fructose, we have the acid and an enzyme. Right? So we don't need to heat because we have the enzyme. We have sucrase. Okay, enzymes there. So there is a function of enzymes. What are cofactors? Sometimes these enzymes require a friend. Right? Without my friend, I'm not going to do my work. Right? Emotional support, right? Some enzymes require some other people, some other things to do their job, right? So those are called cofactors. Okay, these cofactors based on if they are organic or not, right? If they are organic or not, they can be organic. I mean, like a carbon based වෙන්න පුළුවන්, organic, inorganic වෙන්න පුළුවන්, right? So organic cofactors has a special name called enzymes. So your NAD plus is this kind of a coenzyme. Okay, so write down this cofactors of two types as organic and inorganic. Organic cofactors are called coenzymes. There are inorganic cofactors. Usually those are metal ions, right? Zinc 2 plus, Fe 2 plus, copper 2 plus. Those are the inorganic cofactors. Organic cofactors are called coenzymes. We are going to learn a few here: NAD plus, FAD plus, or coenzymes. Okay. මේ page එකේ උතන ඉඳ තියෙනව නේද? Right, other nucleotides other than කියන එකේ මේ side එකේ you have some space. Yeah, online people. Then if you have space, write down underneath cofactors. What are cofactors කියලා? Write down. What are cofactors? Non-proteinous components, non-proteinous components, non-proteinous, right? Non-proteinous components, essential components, essential for the catalytic activities, essential for the catalytic activities of certain enzymes, essential for the catalytic activities of certain enzymes are called cofactors. Are called cofactors. Okay. So you can understand, right? The organic cofactors are called coenzymes. So anyway, your enzyme is a protein, no? Very commonly, enzymes are almost always proteins. Yes. What level of organization of proteins are enzymes? Is it primary, secondary, or tertiary, or quaternary? Okay. Yes. At what level do they get the unique three-dimensional functional? What are the other things? At tertiary level, no? So enzymes are almost all are tertiary structures of proteins. Okay. Something additional. Your RRNA. මේක මත ඉක්මනට අමතක වෙලා ගියත් කමක් නැහැ. Your RRNA. What is RRNA? We just learned. Ribosomal RNA has an enzymatic action as well. Right? It's a very rare situation that something other than a protein acting as a enzyme. Okay, so if you like, under ribosomal RNA, we just learned here in the tute, right, plays an enzymatic role. Write down another function of rRNA. There's one function that provides the site where polypeptide chains are assembled, right? Underneath that, right, has an enzymatic action, for the formation of, peptide bonds. Okay. So he sort of promotes this new bond formation between the amino acids, right? rRNA. Right? Those are some rare MCQ points. Sometimes they will be questioned. They can ask, they can say all enzymes are proteins. Is it true? No. There are rare situations that nucleic acids can be enzymes as well. Okay. So we were talking about NAD plus, no? NAD plus, it's a coenzyme, yes, and electron carrier. How does NAD plus become an electron carrier? When it becomes reduced, right? What is reduction? NAD plus an electron plus an actually there's an like a H plus involvement as well. No need. So it becomes NADH. This NADH is a high energy molecule that carries electrons. We'll learn about the functions of this NADH when we are doing cellular respiration. Okay. For now, like just understand this reaction. It can get an electron. It can become what? Reduced to NADH, right? So the reduced form of NAD plus is called NADH. It acts as an electron carrier. Then you can explain the next function, act as an oxidizing agent. What is an oxidizing agent? Last week we talked about, right? It oxidizes someone else, no? Oxidizing agent ke liye kya hai? It oxidizes someone else. It's an agent of oxidation, right? Yeah, when electron right? That is oxidation. No? When someone is releasing an electron, that is called oxidation. So the oxidizing agent is taking that electron. So what is happening to the oxidizing agent? He is becoming reduced. Oxidizing agent becomes reduced. No? Got it? So this is what happens. Can you see? He's taking the electron and he's becoming reduced. So can you understand how NAD plus acts as an oxidizing agent? Right? If you like, with a pencil, you can write this reaction. This is how NAD plus acts as an electron carrier as well as an oxidizing agent. Online people, any questions? Then the next nucleotide is very similar in sort of behavior and functions to NAD plus as well. There is NADP plus, right? Almost same functions, but NADP plus we can usually find in plants. Okay, NAD plus we can find in animal cells. NADP plus we can find in usually plant cells. So this forms NADPH. So this is nicotinamide adenine dinucleotide phosphate. Okay, it can get an electron and form NADPH. So similar to NAD plus, it acts as an electron carrier. It acts as an oxidizing agent as well. But highlight very importantly, NAD plus is involved in respiration. NADP plus is involved in photosynthesis. Okay, usually photosynthesis is happening in plants. So NADP plus we can usually find in plants. Okay. Finally, FAD. FAD is very similar to these three molecules. All these molecules are involved in like cyclic processes, right? Respiration, photosynthesis, okay, cyclic processes are involved in it. So FAD is actually involved in respiration. Very similar functions: coenzyme, electron carrier, and oxidizing agent. So FAD binds with an electron with electrons to form FADH2. This is flavine adenine dinucleotide. Okay, F is flavine. Okay, no need to know that. So these are the other nucleotides. Any questions? Any questions? Yes, online people. Any questions? No. Green marker is not clear. Yeah, no. But I like my green marker. Okay. NAD plus reacts with electron to form NADH. Okay. What is not clear? This is NADH. So I have to stop using my green marker. DPH. This is FADH2. This is NAD plus. This is NADP plus. This is FAD. Okay, these are electrons. Now, okay. All right. So let's do questions. 10 to next page. 10 to next page. Second MCQ. The number given is 41. Right? Online people, can you see? Which of the following statement regarding nucleotides are correct? Quickly do it. 1 minute. Okay. Online people, this question. This one. Anyone who haven't got the tube? Online people? Everyone has. Okay. Quickly do this. 1 minute. It's a resource book. Is it old one? One Five Brothers or something? Is it an old edition? No, right? Oh yeah. When did he or she do A levels? Last year, then fine. See, that's what I told you. This is the way to go. Like buying Quran. Yeah, a bit old, no? There's only grade 12 one, no? Why is it so big? Because of the use, no? Like it becomes bigger with the use. Others, if you can get it spiral bound, also better. Everyone got resource books? Yes. Kathali? From the you can either download the PDF and get printouts, or what ගත්තේ කොහෙන්ද? That one? NIE එකේ තියෙනවානේ. There's a bookshop, right? From the NIE bookshop, you can buy the resource. There's 2 resource books from grade 12 and grade 30. This 12 one is enough for now. Okay. Okay. let's discuss which of the following statements regarding nucleotides are correct. NADP plus functions as an electron carrier and oxidizing agent. So this is not one single correct answer type question. No, have you seen this kind of MCQs? Have you seen this kind of MCQs? There's a combination of answers. There's a combination of answers. I can't actually remember the combinations, but with time you will actually memorize the combination. So it's like this. You have seen this kind of question, right? If A, B, D are correct, the answer now, like the answer you have to mark in the sort of the answer script is 1. Okay, if A, B, and D are correct, then A, C, D are correct, then it's second answer, right? I mean, like you have to color the second box. If A and B are correct, it's answer number 3. If C and D are correct, it's answer number 4. If any other combination is the correct combination, you have to color the fifth one, right? So, you know, in your MCQ answer script, for example, this is question number 41. No? So, for 41, from question number 40 to 50, you have these kind of questions, A, B, C, D, E kind of answers, right? So, there can be like a combination of answers that is correct, right? So, if what did I say, A, C, A, B, D is correct, then it's answer number 1. Like this, you have 5 circles that you have to color. No, that I use pencil, right? So you have to not color, you have to cross like this. In A levels, you have to cross, right? In our university exams, you have to like color it very nicely, because in our exams there's a machine, right? It's like marked by a MCQ papers are marked by a machine. So in order to be detected by the sensor, you have to color it. But for A levels, they are manually marked. So you just have to cross it. ඔයගොල්ලෝ instruction sheet එකක් හම්බෙනවා හරියට mark කරන්නේ කොහොමද කියලා. Right? නපන්න එපා කියනවා, ටිකෙක්ස් කරන්න එපා කියනවා, and පාට කරන්න එපා කියනවා. You have to just nicely cross it. Okay? So let's move on to this MCQ. NADP plus functions as an electron carrier and oxidizing agent. What do you say? Yes. Electron carrier oxidizing agent? Yes, that is correct. FAD functions as an electron carrier and reducing agent? No, it is also an oxidizing agent. You can cut it and right, you can correct it. Electron carrier part is yes, okay. Then it acts as oxidizing agent. You didn't get the tube. Are you in the group, Puta? Are you in the group? Mithulima, are you in the group? Yes. Okay. Hold on. I'll upload the tube to the group again. So please get it downloaded from the group. Okay. Yeah, I'm sending it. It's a bit large, so it will take some time, but it's in the group. Okay. Okay. Naomi, I put the tute again in the group. Okay. So from next week, I'll start the delivery process. Okay. So this is our final free week. No? Right. So you have already got the tute. Okay. 9 people about it. Okay. So from next week, I'll start. Okay. Second statement: FAD functions as an electron carrier and reducing agent. No, oxidizing agent. Okay. NADP plus and FAD function as coenzymes and electron carriers? Yes, that is correct. NAD plus functions as an electron carrier and oxidizing agent? Yes. NAD plus and NADP plus function as coenzymes and reducing agents? No. Which part is correct? Yes, they are coenzymes, but not reducing agents. They are oxidizing agents. දැන් මෙතනදී දැන් like විනාඩි 5කට කලින් මේ ටික කළා, yes you can do MCQs, right? Because you just learned this. But imagine after 2 years you go to the exam with another ඔයාගේ 2 resource books පාඩම් කරලා යන්නේ. So you will not remember if any of these are coenzymes or any of these are electron carriers. මතක නැහැ. Right? So that's why in the A-level paper, these are questions, right? Small sections. So at this stage, try to get it to your deep memory, right? Not to memorize, just have an idea, right? What is going to happen? All those are sort of positively charged, no? So they can get an electron. They can become reduced. So they are going to oxidize someone else. So they are all oxidizing it. Okay, put a logic එක දැම්ම දාන්න. Don't let the logic be forgotten, right? Memory එක අමතක වෙනවා, but the logic is sort of you something you understood, no? So try to keep it. Okay, that's why what I always say, try to understand in the class, then you will, it's very easy for you to sort of go home and memorize. කොහොමද memorize කරන්න වෙනවනේ, that's the truth නේ, කටපාඩම් කරන්න වෙනවා. But නිකම් blindly කටපාඩම් කරන්න එපා. ඔබ තේරෙන්නේ නැත්නම් then ask again or watch the recording again, right? So what are the answers we got? A is okay, C is okay, D is okay, no? ACD. What's the combination for ACD? Answer number 2. Okay. At this point, no need, just put marks. I mean, put ticks to the correct statements. ACD, කියලා ticks them. Okay. Then this one, like මේ copy cut paste කරලා වගේ තියෙන structure එකක් තියෙන්නේ. What is the structural difference between a nucleotide and a nucleoside? Quickly do it. What is the structural difference between the nucleotide and a nucleoside? Oh, you are not in the group, okay? What's the difference? So they're asking what? The structural difference. So what do you have to say? A nucleotide without phosphate group is called nucleoside. That's it. Nucleotide without a phosphate group. Did we do other questions found in the tube from monosaccharides and disaccharides? No, right? අන්තික කරේ නෑ නේද? These questions? 3 types of lipids found in organisms? No, right? Did we do? We did alone. Okay. අපි කරේ නෑ නේද? ඒ ටික අන්තිමට කරමු, right? As the cool down paper. There's another structured here at the very end of the second page. State 2 common functions of NAD plus, NADP plus, and FAD. This one? Yeah, this question. Online people, can you see? State 2 common functions of NAD plus, NADP plus, and FAD. Do that. 30 seconds. Common functions. There's actually three, no? All are coenzymes, all are electron carriers, all act as oxidizing agents. So, තුනෙන් any two is okay. Yes, am I correct? Those are all three are common functions, right? Coenzymes, electron carriers, and oxidizing agents. Any two of those three is the correct answer. Yes. So, homework in the essay book. Okay, there are two essays at the end, right? So, you need to buy a separate book to write essays. Okay, that's the only book you need to write of your own, right? I give the tools. CR book to write essays and homework. First one, describe the components of nucleotides and explain how nucleotides form the backbone of DNA, and describe the chemical nature and general structure of proteins. These are two separate essays, but not full essays. Right? Can you see it's A, right? There's a B part that I have not included because you can't do. Right? So remember, these are not full essays, right? So it might not have 50 points. 50 points here in the very way, right? Because these are sort of not half, sometimes more than half essay. Right? Because there's part A and B. No? So I have given only the part A. So, but try to write all the points that you can think of, that you think that is relevant to the topic, right? Essay එකට ඕනේ කියලා හිතන points ඔක්කොම ටික ලියන්න. Don't think it's a half essay in the points 25 ලියන්න ඕනේ. එහෙම don't think like that, right? When an essay has two parts, it's not equally divided, right? ඒ කියන්නේ essay එකට part A and B තිබ්බා කියලා of the total 50 points 25 යන්නේ. Sometimes it can be 30-20, right? Like that. Okay, sometimes it can be 35-15. It depends. So one part can be a big topic and the second part can be a small topic, like that. So අපිට අමතක වෙන්න කලින් let's mark that water essay. Like I think I gave it on the second week, no? Water essay එක ලිව්වද? Did you get an essay book at least? Yeah? Essay ලියන්න පොතක් ගත්තා? Yeah. Water essay එක ලිව්වා? Okay? Yes or no? Yeah. Okay, take it out. I don't mind, but you have to sort of, you are independent, so you have to sort of do your responsible for your own work, okay? Right, okay, you have written. You were not there when I gave them. So go home and write, okay? Today I'll give the marking scheme. Yeah, nice. It's fine. Yeah, you are new. And yeah, everyone else has written, no? So let's mark it. What was the topic? Physical and chemical properties of water important for life. That is the topic, no? So let's try to sort of make a marking scheme ourselves. So, you know, in future, you don't know, I mean, you don't have past paper marking schemes for every topic, no? Right? So you have to write essays of your own. It's part of your studying process, right? For example, in future, you just studied about the excretory system, right? Process of urine formation. So one of the best ways to study that is to write an essay. You just imagine this is an exam question. They are asking you describe the process of urine formation. So you write an essay. Then you just mark it yourself. You think, this could be an essay point. Then you give yourself a mark. This was a mark. This was a mark. Right? It's one of the most effective ways to study. Right? Then, for example, I gave you how nucleotides form the backbone. So if you just finish studying now, one of the best ways to sort of test yourself and also to actively remember that is to write an essay. Okay. So use essay writing as a study tool. That's what I'm trying to say. Use essay writing as a study tool. that's going to benefit you so much. නිකන් පොත දිහා බලන් highlight කරා කියලා ආයි නිකන් පොත දිහා භාවනා කරන්න වගේ බලන් හිටියා කියන එකට වඩා doing something active, actively doing something, writing something from your memory, recalling it and writing it, then by yourself checking it again, it's all going to be so valuable. These skills, right? Self study තමයි අන්තිමට ඉතුරු වෙන්නේ. Okay, class එකෙන් කොච්චරව කියලා දුන්නා if you haven't nailed the self study part, it's going to be a problem, especially not for bio sometimes, but for physics වගේ subjects වලට if you can't go home and do it by yourself, that's going to be a problem, නේ. physics class එකේ පොතල පෙව්වත් වැඩක් නැහැ if you can't do it by yourself. අයෝලත් එහෙම component එකක් තියෙනවා, that is the essay writing, right? No matter how much I teach you theory, but if you can't write a proper essay with your own practice, that's a problem, no? So දැන් මේ ඉඳලා ඒක පුරුදු. Right. Okay. Let's try to mark our water essay. So how do you get the online people? Have you written the essays? Online people, have you written? Okay. Right. So I'm going to read out the marking scheme, and when I say a point, give yourself a mark, right? But if I say without a certain, if a certain word is missing, do not take marks, right? May word එක නැත්තම් marks දෙන්න බෑ කිව්වොත් you don't give yourself a mark. Okay? Right. So the very first point you have to write is water is a vital inorganic molecule. Okay? You don't have the resource book, right? You have? Yeah, turn it. This point, water is a vital inorganic molecule. So if the word inorganic is missing, no marks. Yes. Turn to water. Yeah. From here onwards, right? So actually, if you stick to the resource book, it's the best way to go. But have some sort of a general idea as well. Otherwise, if you remember completely අමතක වුණොත් මොනවාදම තියෙන්නේ කියලා නිකන් අර පාඩම් කරන්න ගිහිල්ලා, right? But if you have some sort of idea, you can write something. So try to have the understanding first, then try to memorize. Okay. So first point, water is a vital inorganic molecule. If the word inorganic is missing, no points. Okay. Anitya life could not exist on this planet. Then water is important for the following reasons. Then write vital chemical constituent of the living cell. 1 mark. Okay, point. If the chemical word chemical is missing, then no points. Okay, it has to be a chemical constituent. Okay. The next point provides a biological medium for all organisms. 1 mark, right? Biological medium is a keyword, like a section of words you can't miss. Okay, biological medium. Moving on, you have to draw the water molecule, right? You have to draw the water molecule. How do you draw the water molecule? I'm giving you 4 marks for the diagram of the water molecule, right? Oxygen, hydrogens, slightly negative charge, slightly positive charge, and the angle. 107 point, what is it? 0.5, 104.5, sorry. This angle, this water molecule gives you 4 marks. Okay. You have to draw this. Okay. Right. Moving on. Next point. Water is a small polar angular molecule. Next statement you have to write. Why වචන තුනට small polar angular එකකට එක ගානේ 3 marks. Okay. Small polar angular molecule. Each word 1 mark. 3 points. right? This can change, but what I'm saying is these are the things that you should not, I mean, you should be careful not to miss, right? The makeup, the marking scheme can slightly change, no? Because different people are making it, no? But what I'm trying to give you is these are the things that you cannot miss, right? So you have to train yourself. These are the important points I need to write, okay? Then, so small polar angular 3 marks. Polarity is an uneven charge distribution. Usually those things are given. So in a water molecule, oxygen atom is slightly negative. For saying slightly negative, 1 mark. And the hydrogen atom is slightly positive, 1 mark. Then the definition of hydrogen bonds, very, very important. Weak attraction between slightly polar hydrogen atom of one water and the slightly polar oxygen of adjacent water molecule are known as hydrogen bonds, right? So you cannot miss weak attractions, right? Slightly polar you cannot miss, and the adjacent water molecules you cannot miss. Like a segment of words නැත්තම් you cannot get the point. Okay, what are hydrogen bonds කියන එක definition එක. It has to be very precise. Okay, it has to be very precise. Definitions ලියනකොට no excuses. Okay, then you have another diagram that is the hydrogen bonding, no? That diagram with the four water molecules and the hydrogen bonds between slightly positive and slightly negative. Yeah, can you see that diagram? For that diagram, like I'm not going to draw it. For that diagram, I'm going to give 2 marks. For this one, 4 marks, right? If you have drawn that diagram, I'm going to give you 2 marks, but no marks if you haven't labeled the hydrogen bonds, right? Hydrogen bonds වලට arrow එකක් දාලා hydrogen bonds කියලා label කරලා නැත්නම් no marks. Okay, 2 marks දෙකම නැහැ. Okay, marks දෙකම නැහැ from the diagram if you haven't labeled the hydrogen bonds. Then okay, properties of water arise due to hydrogen bonds. When the water is in liquid form, the hydrogen bonds are very fragile. That's important, right? Hydrogen bonds are very fragile. 1 mark. Right? Otherwise, if the hydrogen bonds are very strong, water would be like a solid in liquid in the room temperature. No? But they are very fragile. That's why water exists as a liquid in room temperature. So for saying H bonds are very fragile, 1 mark. Then moving on to the 4 properties of water. For each property 1 mark. ඔගොල්ලෝ මුලින්ම එක list එකක් විදිහට ලියලා තියෙන්නත් පුළුවන්. Or you can have written as at the start of each paragraph, නේද? Right? So cohesive behavior කියලා ලියලා if you have started a paragraph, then if you have written ability to moderate temperature and written a paragraph, it's fine, right? At the start of the paragraph or at the beginning as a list, if you have written the 4 properties, then 1 mark each for the 4 properties, 4 marks. Okay, cohesive behavior, ability to moderate temperature, expansion upon freezing, versatility as a solvent. ඔය හතරට marks 4. Then cohesive behavior, the definition of cohesion. Attraction between water molecules due to hydrogen bonding is known as cohesion. 2 marks there, right? For the word cohesion, 1 mark, and attraction between water molecules කියන එකට 1 mark, right? That is the describing of the cohesion. No? You have to say it is the attraction between water molecules. Okay? But if you haven't written attraction between water molecules and written something else and have written cohesion, එතකොට ලකුණු දෙකම නැහැ. You have to get it, right? If you have confused what is cohesion, cohesion කියන්නේ water attraction එකනේ. So if you have written something else and then written cohesion mark එකක් නැහැ. Get it? You follow, right? Cohesion කියන එක නිකන් ලියලා හරියන්නේ නැහැ. You have to write what is cohesion correctly. Yeah. So 2 marks for that one sentence. Then next, addition between water and other substances, 1 mark, is known as addition, another mark. If you have written what is addition incorrectly, no marks. So next, due to cohesion between water molecules, water and dissolved substances such as, so water and dissolved substances, 1 mark. Kawuda transport in water and dissolved substances, 1 mark. Such as minerals are transported as a continuous column through xylem against gravity. The word xylem is going to get you a mark. Okay, the word xylem against gravity. Addition between water molecules and cell walls also help in conduction of water and dissolved substances. So water molecules and cell walls, 1 mark, and conduction of water and dissolved substances, 1 mark. Then water has high surface tension, 1 mark. Next point, water has high surface tension, 1 mark. Next point, in an aquatic system, upper surface water molecules are attracted by lower surface molecules. So for saying upper surface, 1 mark, lower surface, 1 mark. And it forms a water film. Water film is a keyword, so 1 mark. Water film. Water film. In the example, water skaters can walk on the surface. Water skaters both 1 mark. Right? You have seen those insects, no? Very light ones that can walk on surface of a pond. Yes, water skaters. Then next one, ability to moderate temperature. So you have written it here or earlier, 1 mark, but the part didn't, right? Ability to moderate temperature. This actually work okay, only 1 mark. So what is the definition? Water can absorb or release a relatively high amount of heat energy by a slight change in on temperature. So for saying relatively high amount of energy, 1 mark, and with a slight change in temperature, 1 mark. So that sentence, 2 marks. Okay. Then due to high specific heat, 1 mark. High specific heat, 1 mark. Water will function as a thermal buffer. Very, very important word. Thermal buffer, 1 mark. Next point. Due to high heat of vaporization, 1 mark. Due to high heat of vaporization, 1 mark. With minimum loss of water. Minimum loss of water, 1 mark. An organism can release much heat energy. Okay. Then this helps organisms to cool their body surface. Body surface, 1 mark. Cool their body surface. Right? Just saying cool will not give you the mark. You have to say it's the body surface. Okay. No, you can't give examples from outside the resource book. Right? You cannot give examples from outside the resource book. That's the reality. Okay. All right. So there's no such freedom. It's not like any other exam. Okay. Body surface, and the example is prevent from overheating. Prevent from overheating. The word overheating is going to give you the mark. Prevent from overheating. Then evaporation of sweat from human skin, 1 mark. Helps to maintain body temperature. That whole statement should be there, but you cannot miss the word sweat from the skin. Okay. No marks. Evaporation of water from skin over it has to be sweat, right? Transpiration in plants to help to cool the plant body surface and prevent from becoming too warm in the sunlight. Right? 1 mark. But if you have missed the word transpiration, no marks. Transpiration කියන වචනේ නැත්තම් no marks. Okay. Moving on, expansion upon freezing. So you have to say increase in temperature of a substance reduces the density 1 mark. Increasing temperature reduces density 1 mark. Then on the other idea, decrease in temperature increases density 1 mark. But if you have missed the word density, no marks. Okay, density is a keyword. Density clearly under. Okay, right. When the temperature of water falls below 4 degrees Celsius, 4 degrees Celsius, 1 mark. That value is a very, very important value. It begins to freeze and forms crystalline lattice called ice cubes. Crystalline lattice is a keyword. Okay, crystalline lattice you have to have written. Crystalline lattice. So then next point, water has maximum density as 4 degrees Celsius, 1 mark. You have to say maximum density. You can't say maximum weight or nothing else. Okay, maximum density has to be there. Hence, hence කියන්න ඕනේ ice floats on water. Hence නෙවෙයි chicken හරි ice floats on water කියන idea එක තියෙන්න ඕනේ, right? Don't pay that much attention to the English, right? Just give the idea. Ice floats on water කියවනම් හරි. So then it's an important property of water in polar regions where organisms in aquatic bodies can survive during the winter. So that idea 1 mark, but what do you have to, what is the keyword? Anyone? What is the keyword in the statement? Polar bodies, right? You have to say it's in the polar regions. Sorry, polar regions, polar regions. Okay, moving on to the final property. Versatility as a solvent. This is given to water due to its polarity, 1 mark. This is given to water due to its polarity, 1 mark. Polar molecules and the example. Polar molecules, example, glucose. All together 1 mark, okay. Ionic compounds, example, NaCl, 1 mark. Molecules with both polar and ionic regions, example, lysozymes, 1 mark. Can dissolve in water, so that's the statement. Then final point, water molecules surround each of the solute molecules and form hydrogen bonds. What point cannot, what is the important point you can't miss? Hydrogen bonds, right? That is how these things dissolve in water when water forms hydrogen bonds around them. No? I counted 48 wage points. Count how many you got online, people. I want you to put the number. How many points? I think I got 48. Right? Count how many points you got. Yes, this is a nice diagram. Right? This is how you have to draw. At least 3 molecules are when you are drawing the hydrogen bonds. Right? At least 3 were thrown. Yeah. Online people, how many out of 48? If you are shy, you can send to me personally, okay? This is the very first essay, right? So it's an important, let's say, milestone, right? People who haven't written, please write again, okay? Not write again, write. And if you have got below maybe 40 or something, better if you write again, okay? This is a very straightforward topic. Yes. check it out. Okay, fine. Right again. Okay. Nice. Better right again. Okay. Not bad. Right. To improve handwriting technique, right? Not. I mean, it's not that bad. Your handwriting is okay. This is exceptional. Printed buggy, right? So yeah. All right. 38. Yes. Yes. Yeah. Good. Okay. So first essay, if you feel like you can do better, then you get 50 points. I mean, like counted 50 points. I gave. Yeah. I have given 50 points. Lu. Okay. Online people are bit better. 44, 40, maybe gathering in the done, right? Points with the diagram 48, right? Yeah. I thought I got 48. For this diagram 4 and for the second diagram 2 points. Okay. Yeah. I think that's enough. For the future essays, I'll be giving the marking scheme. Okay. Future essays, time around the 20 minutes. Right. I won't be discussing this much detail, just going through the marking scheme. I'll be giving marking schemes. Okay. Hari. Any questions? Very good if you have got above 40. Okay. Okay. Moving on. So it's, yeah. Let's start cellular basis today, without further ado, as they say. So we have finished microscopes. So from this point onwards in your tube, there is no resource book. Resource book, right? It's only just diagrams. So when I tell to turn to this page in the resource book, you have to have the resource book by the side. Okay. So take out the resource book, page number 24. Anyone who haven't got? Can you share it? Put resource book. You have got, yeah. Anyone else? Everyone has got, right? Yeah. So page number 24. Online people also, please, if you have a PDF of the resource or whatever, come to page number 24. Historical background of the cell and structure and functions of subcellular units. Okay. So first, we are going to do some history. That is how this concept of a cell and also the study of cell came to be. Right. That is called the cell theory. The cell theory was actually presented by 3 people. Right. That those are given in page number 24. You have Matthias Leiden, Theodor Schwann, and Rudolf Virchow. 3 people. Right? Before that, there's a little bit of history. Let's read it. All organisms are composed of cells, right? Either one cell or many cells. What do you call one cell organism? A unicellular organism, right? Organisms can be unicellular, multicellular, right? So the basic unit which can be called living is the cell, which may be a single cell organism, such as Chlamydomonas. Have you heard Chlamydomonas? It's a protozoan. Okay, single cell organism that you can see in pond water, right? Chlamydomonas, protozoa. Yeast. Anybody know what yeast is? What are the yeast use? Bakery. And what is bakery products? What is yeast? Even more than that, alcohol production, ethanol, right? Fermentation yeast, right? That rahadad naturally gahedima yeast. In the wind you have yeast floating, right? It gets naturally deposited and it turns the sugar in the what? It will right? It turns that into alcohol. But in these massive factories, you add the yeast and to allow the fermentation to happen. Your microbiology, right? You have to do the applications of microorganisms. So yeast is a fungus. Okay. Saccharomyces cerevisiae, right? Yeast is a single cell fungus. Okay. So multicellular, there are plants and animals. Whatever the kind of animal, how many cells do they, no matter how many cells they have, the cell is the basic structural and functional unit. That is, you cannot forget. No? So these are the things. Any stage below the level of a cell cannot be considered living, whether it's a single cell organism or multicellular plant or animal. Right. Then Robert Hooke, 1665, examined a cork using a simple microscope, gave the term cell to describe the units. Highlight. Who gave the name to the cell? Robert Hooke. Then Anton van Leeuwenhoek, which is a contemporary means sort of Samakalina at the same time. He was the first to describe and record living single-celled organisms such as Euglena and some bacteria. Okay. So here in the tube, you have the Robert Hooke's microscope and the cork sample he observed. You can write in front of it. Cork cells, C-O-R-K, okay, again, right? Cork cells observed by Robert Hooke. Cork cells observed by Robert Hooke. Okay. Brilliant. Moving on, so Anton van Leeuwenhoek, he described and recorded living single-celled organisms. Then the like the three people who got together to present the cell theory. Matthias Leiden, Theodor Schwann, and Rudolf Virchow. No? Those three people had sort of got together all their discoveries. So Leiden was a botanist. He studied plant cells, and he said all plants are made of cells. Theodor Schwann was a zoologist. He was studying animal tissues. So animal cells are also composed of this cell. Rudolf Virchow, he's talked about a bit of cell division, right? He said all cells arise from previous cells, right? So ඔය තුන් දෙනාගේ discoveries තුන එකට එකතු කළා we have our modern cell theory. ඔය cell theory එකේ තියෙන points තුන I think memorize කරන්න very very important. All organisms are composed of one or more cells. The basic structural and functional unit of organism is the cell. All cells arise from pre-existing cells. Highlight them. Highlight. Very very important. Okay. So moving on to the organization of cells. So in our world, we can classify cells based on a certain level of organization, right? Basic thing that we use to classify this is how organized the nucleus is, right? Nucleus it's like the main controlling center. So we determine if this cell is this kind and this kind by looking at the nucleus, because nucleus is the most important part, right? So if a cell doesn't have a properly organized nucleus, we call it a prokaryotic cell. Okay. If it has a nicely organized nucleus, we can call it a eukaryotic cell. So by evolution, your eukaryotic cells are usually better than prokaryotic cells. Usually prokaryotic cells are all unicellular and usually microscopic, right? ඇහැට පේන්නේ නැහැ prokaryotic cells usually. We are usually composed of eukaryotic cells, and usually eukaryotic cells form multicellular organisms. That is the logic. But doesn't matter what kind of cell it's prokaryotic, the eukaryotic කියන එක අදාළ නැහැ. We have 4 features that are unique to all cells. So not unique, common to all cells. What are they? Please read. All cells are bounded by a plasma membrane, which is a selective barrier. ඕනම cell එකකට plasma membrane එකක් තියෙනවා. Highlight the word plasma membrane. All cells have a plasma membrane. Within the cell, there's a semi-fluid jelly-like substance called the cytosol. Highlight the word cytosol. Subcellular components are suspended in the cytosol. So that statement has two things, right? හැම cell එකක් ඇතුළෙම there's a jelly-like substance called cytosol. And in that cytosol, there are subcellular components. So what does that mean? All cells have some kind of subcellular components as well, right? It can be a very simple cell, it can be a very complicated cell, but doesn't matter. All have මොනා හරි subcellular components තියෙනවා, right? Third one, they carry DNA as genetic material. All of their information will only be stored in DNA. There is no other molecule involved in storing information. ඕනම living cell එකකට DNA තියෙනවම තමයි. Okay? All living cells have DNA. The ribosomes are found in all living cells. ඕනම cell එකක ribosomes තියෙනවා. Because why? Ribosomes produce proteins, right? Proteins is essential for all cells. Okay? So in your tube, just to memorize a bit better, you can't see that clearly. I haven't drawn an arrow to the ribosomes, but can you see in the cover page? This is what? This is a bacterial cell, right? Bacterial cell, there are some blank boxes that you can fill. Okay, the blue color layer, the one after. Okay, before that, I'll show you some cells with the projector. Okay. Then after that, we can label this. So those 4 features of all cells are very commonly questioned in the MCQs as well as in the structure. What are the features you can see in all living cells? Okay. So if I were to show you what is this prokaryotic and eukaryotic cellular organization, this is a prokaryotic cell. This is its DNA. It's just arranged just everywhere in the cell, nicely packed into a nucleus. Okay, it's just නිකන් නූල් බෝලයක් වගේ, right? It's like a bunch of DNA, not nicely arranged. Okay, but if you take a animal cell and if you remove this sort of area and it's surrounded by some membranes, can you see this purple color ball is your nucleus, right? So it has a nice nucleus, and if we remove it, inside you can see the DNA, right? Can you see? So proper organization, nice organization is there in any eukaryotic cell. So moving on to the features that you can see, what is, so in a bacterial cell, there's this outermost covering, you don't need that at this stage, I'll explain later. But inner to that, we have this, sorry, this one, that is called the plasma membrane. Can you see? All cells have a plasma membrane. All cells have a plasma membrane. If you take an animal cell, this is the plasma membrane, right? So, can you see? This is the plasma membrane. And then what else? So, this is the jelly-like material, that is the cytosol, right? It's completely filling the interior of the cell, jelly-like material, right? And suspended in it, you have so many subcellular components, right? So, this is a Golgi. These are, I think, mitochondria. This is what, it's a peroxisome. This is smooth endoplasmic reticulum. This is rough endoplasmic reticulum, right? So many subcellular components are there. Okay. Then what was next? All have DNA, no? So that's, you saw, no? Inside the nucleus you have DNA. Then finally what? All have ribosomes. Can you see these small, not those, these dots everywhere? These are ribosomes, right? Arthing? Rough endoplasmic reticulum attached. This. These are ribosomes. All living cells have ribosomes. Okay. So this is the same diagram that I have given in your tube. You can label, right? This is called the capsule, right? Have I drawn an arrow to that? Yes. The outermost green color. I'll explain this. This is it's called the capsule. Okay. Write the name in the box, right? Capsule. Follow the arrow and the capsule. Then the second layer is the cell wall. Third layer, plasma membrane. Then this jelly, cytosol. What else? Then because this is what kind of cell? This is not a advanced cell, no? This is a prokaryotic cell. So as I told you, they don't have a nucleus. Instead, they have what we call as a nucleoid. Okay, nucleoid. Label that as a nucleoid. No nucleus, just a nucleoid. And can you see these small dots like things? What are those? Ribosomes. Ribosomes. And this one, this big thing, like a tail, is called a flagellum. Flagellum, right? It's used to move, for the cell to move. Okay, flagellum. Flagellum. This is what? Nucleoid. Okay. Good. Moving on. So there's a chart in page number 25. Page number 25 of the resource book, there's a chart. This chart is very, very important. Okay, page number 25, and it extends to page number 26 as well. So that chart I'll come back after we have completed the list. You will not understand, but it will be sort of waste of time. So after you have studied the lesson, I'll come back and explain this chart. It's like a summary that should be at the end of the lesson. And I have it's at the end of the tutor as well, right? So you can just study it, right? There are so many MCQ points in this chart. Right? There's my face in it also, but don't mind that. Right? So just study the chart, okay? That's why I have given it. So this has so many MCQ points, okay? So have it, and I refer it again and again and again, okay? And so we have time to talk about the plasma membrane, yes. What do you say? Do we do the questions from biological molecules, or do we discuss the plasma membrane? What do you say? Okay, අන්තිමට තියෙන questions ඒක අපි next week කරමු නේද questions එක. අද plasma membrane එක ගැන කතා කරමු නේ. Yeah. Okay. So and in the tute here I have given you some cells. These are some cells. Can you online people see? What kind of a cell is this? Here you can say it's a gamete, like specifically what kind? It's a sperm. Yes, it's a male gamete, no? It's a male gamete. You can label it, it's a sperm. Then what's the second one? එයාගේ counterpart. What is it? It's an egg. It's an ovum. You can say it's an ovum or an egg cell. Right? So why did I give those? Actually, your sperm is actually the smallest cell in the body. Smallest cell in the human body. Okay? And your ovum is actually the largest cell in the human body. Right? So during fertilization, I will show you later when we are doing reproduction. I mean studying reproduction. A single egg cell will be surrounded by thousands of sperms. Okay? So that's how big the egg is and small the sperm is. Right? There's a massive diversity. Then finally, what is the other cell that is given here? It's a neuron. Yes, a nerve cell. A neuron. Yeah, very good. Nerve cells are so important. I mean, so special. You only have the neurons that you were born with. ඉපදෙද්දී තිබ්බ neurons ටික විතරයි තාම තියෙන්නේ. Right? අපේ muscle grow වුණාට, height එක, bones දිග වුණාට, no matter how much we grew, we only have the neurons that we were born with. Right? Brain එකේ, spinal cord එකේ, body එක පුරාම we only have the nerves that we are born with. So they have grown a little bit, but the number is the same. Okay? So nerves are actually, nerve cells can be very long. They are the very longest type of cell in the body. And also they can't be regenerated. At birth, so due to some like environmental factors like smoking, alcohol, right? Those kind of things can destroy nerve cells, right? And also some diseases like diabetes can damage nerves. No? Diabetes, have you seen their feet have wounds, right? Hey, diabetic people, have you seen? Why is that? because they don't feel their legs, right? They can't feel their feet. So they walk on නිකන් ගල් උඩ ඇවිදගෙන යනවා and දෙකොල්ලෝ නිකන් මෙහෙම කකුල වදිනවා කොහේ හරි. They don't even feel. So දෙකොල්ලෝ දන්නේ නැතුවම damage වෙලා. Right? So they have unknowingly they have damaged their foot. Right? So they have ulcers. And the other thing is because they have high blood sugar, ඒගොල්ලන්ගේ තුවාල හොඳ වෙන්නේ. Right? So unintentionally they have wounds, and also because of the high sugar, their wounds don't heal. And because of the high sugar, හොඳට bacteria හැදෙනවා wounds වල. Right? Nicely නිකන් culture medium එකක් වගේ ලස්සනට bacteria හැදෙනවා in diabetic wounds. Okay? So what I said is those kind of diseases can also damage your nerves, but there is the nerves will not grow back. Right? You cannot regenerate them. Like if you have a wound in your skin, හැම නිකන් layer එකක්ම ගැලෙවුවොත් it will grow back, no? Because your skin is not a permanent tissue, right? It can regenerate like that. Okay? Liver එකෙන් කැල්ලක් ගත්තොත් you can donate someone part of your liver, right? It will grow back. Your liver. It has the ability to regenerate. Or pancreas එකෙන් කැල්ලක් දෙන්න පුළුවන්. අග්නාශ. කැල්ලක් දෙන්න පුළුවන්. You can donate it. It can your pancreas will regenerate. But nerves don't have that ability. Okay? Just something addition. I think I want to talk a little detail about plasma membrane. So let's do it next week. Let's do questions. Okay? Let's do questions. Did we do any of the MCQs given here? Did you do any MCQs given here? Online people? Did we discuss any MCQs given in the tute? Like except the one we did today. Others discuss kare nada. Then 5 minutes. Do this first page, all the MCQs and the structure. First page. 5 minutes. Okay? This page. Okay? Try your best. Okay? Online people, this page. Okay? In the tute. 5 minutes. Next page. Okay, shall we discuss? Yeah. I think 4 MCQs and few structured, no? Easy, right? We have studied what we did the previous weeks. Yeah. First one, online people. Which of the following statement is correct regarding carbohydrates? Very first MCQ. We didn't do this, right? No? Not? Yeah. Condensation reaction between two glucose molecules from a non-reducing sugar molecule. What does two glucose molecules produce? Yes. Glucose there, maltose. So is maltose a non-reducing sugar? No, it's a reducing sugar. So you can write a review. You can say glucose is maltose. Write down the reaction glucose plus glucose is maltose. And in front of it, right, it's a reducing sugar. You can say G plus G is maltose, right? It's a reducing sugar, maltose. The only non-reducing sugar we know is sucrose, formed by glucose and fructose. Second one, galactose is a ketose sugar. No, right? Is it? No, right? Yeah, glucose and galactose are the aldose sugars, right? Yeah, fructose is a ketose sugar, no? So galactose you can write. Galactose is a aldehyde sugar, no? Write a review. Galactose is a aldehyde sugar. Galactose is an aldehyde sugar. Yeah. Or you can say it's an aldose, right? Aldehyde sugar. Similar thing is aldose, right? Glucosamine is the monomer of storage components in fungi. Storage monomer. Yes. Monomer storage component. Sorry. What is the storage component in fungi? What do we store? Store, okay. Store in fungi is glycogen. Glycogen is composed of what? Glucose. Right? So, confused කරනවද? කෙලිම fungi ගැන හිතුවාම මොකක්ද මුලින්ම ඔයගොල්ලෝ හිතන්නේ? Chitin. Right? Chitin නම් yes. Glucosamine, right? So you're going to put a tick, right? You're going to take it as correct. But what they are asking? Storage. Storage is glycogen, no? So it's wrong, right? You can say, you can write in front of it, storage component is glycogen. Glycogen is composed of glucose. You can write glucosamine is the monomer of chitin, which is glucosamine is the monomer of chitin, which is a structural polysaccharide. Not a storage component, right? Galacturonic acid is the monomer of a structural polysaccharide in plants. Galacturonic acid is part of what? Pectin, no? Pectin is part of middle lamella. So, is a monomer of a structural polysaccharide in plants. Pectin. We take it as structural, right? Pectin is structural, right? Yeah. Yes. That's okay. Hemicellulose is a branch polysaccharide made of trioses and pentoses. Hemicellulose. No, that's wrong, right? What is the composition of the monomers of hemicellulose? Pentoses and hexoses, right? you can correct, you can cut the triose and write what? Pentoses and hexoses. Yeah, that's your review. Okay, so we did the second MCQ. Then third one, which of the following statements regarding lipids is correct? First statement, lipids are macromolecules consisting of carbon, hydrogen and oxygen. Sorry? Sorry? We did it. Okay, we did it. Right? We did, right? Everyone has written a review. Okay. Then next one, some nucleotides. Okay, some nucleotides. We didn't do that. Right? We some nucleotides. Fourth MCQ. Like the number is 2. Okay. Contain hexose sugars. Is there any nucleotide containing hexoses? No. Always it's a pentose sugar, no? So you can cut it and say contain pentose sugars. Act as organic cofactors? Yes. What is the other name for organic cofactors? Right in front. Coenzymes. Then third one, serve as enzymes. Coenzymes. Enzymes but are they enzymes? No. They are just helping the enzyme. So they are never enzymes. They are just coenzymes. Okay. Act as oxygen carriers. No. Right? What are they carriers of? Electrons. Right? They carry electrons. You can correct it. They are not oxygen carriers. They are electron carriers. They serve as food reserves? No. Some nucleic acids අපි කැම විදිහට storage substance නෙවෙයි. What is the best storage substances? Either lipids or polysaccharides, right? Those are the best storage substances. So what's the answer? Act as organic cofactors. Number 2, no? Proteins, okay? Online people following, right? Any questions you can ask? Form the secondary structure due to disulfide bonds. Secondary structures of proteins are formed due to what? What is the secondary structure? Either alpha helix, beta pleated sheets. Those are formed due to what? What bonds? Hydrogen bonds. But specifically, what kind of hydrogen bonds? Yes. Within the molecule, hydrogen bonds, right? Yes, within the molecule. Intramolecular hydrogen bonds between the oxygen of the carboxyl and the hydrogen of the amino. Write down. Cut the disulfide bonds and say proteins form the secondary structure due to intramolecular hydrogen bonds. Intramolecular hydrogen bonds. Online people, first statement, form the secondary structure due to intramolecular hydrogen bonds. Okay. Second, made up of 26 different amino acids. How many? 20. Cut the 26 and say 20. Third one, composed of carbon, hydrogen, oxygen, nitrogen, sulfur and phosphorus. Usually phosphorus is not always not there, right? Only sulfur is, we stop at sulfur, right? If you like, cut the phosphorus, phosphorus in proteins. But there are some amino acids with phosphorus, but not always. Okay. Would not be denatured by detergents. They are, right? It's one of the right, agents of denaturation is detergents, no? Detergents. Okay. So That is wrong, right? They are denatured by detergents. Contribute to transport of materials. There are transport proteins. Give me some examples for transport proteins. Who transports oxygen and carbon dioxide? Hemoglobin, right? And any other thing? Who transports fatty acids? Serum albumin, right? Serum albumin transports fatty acids. So, what is the correct statement? 5, no? Contribute to transport of materials. Okay, first structure, name two monosaccharides that form a non-reducing disaccharide. Non-reducing disaccharide, what are they asking about? Sucrose, no? What is the non-reducing disaccharide? Sucrose. What are the monomers of sucrose? Glucose and fructose. Next structure we did, okay? Then the next question, 1A, okay? Name the 3 major types of lipids found in organisms. Did we do this? We did this, no? Did we do part B? Okay, I'll just go through what are the 3 types of lipids: fats, phospholipids, and steroids. What is the type of lipid that forms a major component of the cell membrane? Phospholipid. What is the main structural difference between saturated fatty acids and unsaturated fatty acids? You also did this, right? Saturated fatty acids only have single bonds in the hydrocarbon chain, right? And unsaturated fatty acids have one or more double bonds. Yes? Okay, right? So next week we can start plasma membrane, right? I'll try to finish plasma membrane and the nucleus. Hopefully half of the all the organelles. We have to go a little bit fast, now we are a little bit behind. So, but doesn't matter, we are doing the questions now. So I hope you come study it. Like after we do anything, you have to study, right? Otherwise, there's no use of doing the questions, right? Questions if you don't study. So that's why I expect from you to have an idea to do the questions with me at the class. That's what I expect, right? So next week will be the new class of the month, and yeah. So if you have to, I think, pay admission, and if you are not an old student of ISM, I think you have to pay the admission and stuff, right? So you can talk to the I office about that, right? And so for online, this is for online people. The online class, the Zoom link and the recordings will be available. This sort of a website that is called Gurutaru Online. Okay. So I'll send all the details. You have to enroll to that website, and through that website, the tube delivery will be done free of charge, and the fees payment, either card or I think bank transfers or whatever you have to do to that website. I won't be handling any of the online issues. There are separate people to do that. I'll send the phone numbers of those people, but you can ask anytime anything related to the academic stuff. Okay, any theories or like subject matters you can ask, but I'll send the technical people who are handling the website to you, so you can solve all the problems with enrollment, delivery, fees payment, everything. Okay. Any questions? Okay, so for you guys, I'll be creating anyone. So I have this list. Anyone whose number is not here? Yeah, you can put your number as well. For the permanent students of the physical class, I'll have a group. So I'll sometimes be able to put the recording. Right? If you have missed, I can request to me personally. I'll be able to send the recording on the basis that you don't share it with anyone. Right? But don't miss classes. Right? Better to join the physical classes. Right? And yeah. Anyone else? So yeah, online students, any questions about the enrollment process? I'll send all the details. So through this website, the Zoom link and the recordings will be accessible. Okay. Yeah, any question? Okay. Then if you don't have any questions, you can go. Online people, any questions? Any question? No. Maybe next month school start करेंगे. Multi is right? 80% by input below 50% of input is going to be a problem, right? 760 is going to be fine. So try to have some kind of a practical velocity and okay, right. Any questions? Okay, thank you. Okay, you're welcome. Okay, you can go.
Recording stopped.
Fine. So I'll give you the two. Okay. Yes.